Very good evening, everybody. Good afternoon, everybody. So today, myself, Dr. Hemani, and I have this opportunity to welcome you all, the dignitaries, the academicians, professionals, and all the other attendees to this conference on recent advances in agriculture, food security, veterinary, and allied sciences for livelihood and environmental sustainability. This conference is being organized by Atal Bihari Vajpayee Vishwavidyalaya Bilaspur, University of Agriculture Sciences Raichur, Sant Gahira Guru Vishwavidyalaya Sarguja, Vital Biotech and Agriculture Forum for Technical Education of Farming Society Kota. So, first of all, I would like to congratulate you all to be a part of this prestigious international conference and to be able to share a virtual podium along with such honorable dignitaries. So first of all, without wasting much time, I would like to invite Dr. Sumitra Tiwari, Assistant Professor, Atal Bihari Vajpayee Vishwavidyalaya Bilaspur. Sir is also an organizing secretary of this conference. So, sir, I would like to invite you to provide all of us an overview of this conference. So, over to you, sir. Who, sir, has the rights? Yeah,
Yes. It is right now unmute. So,
thank you, sir.
Namaste to all of you. Good afternoon, everybody. Is I audible?
Yes, sir,
Audible.
It is my absolute pleasure to welcome you all, delegates, academicians, and participants in 4th International Conference on recent advances in agriculture, food security, veterinary, and allied sciences for livelihood and environmental sustainability. We are witness to transform era of profitable agriculture, innovation in technology. This boosts our economy, provide employment and means of livelihood beyond gender, language, and religion, keeping in mind sustainable development goals and food security. This conference will enlighten all of us in this area. That is a great pleasure to us because we have a great dignitary. Our presence is greatly appreciated and truly means a lot. First, we start with our Vice Chancellor, Professor Adrian Vajpayee. He is not able to join due to some technical issues because he is gone Pune, Maharashtra, and their brief and their thought we can hear after some another program. And we also admire, we have 5 vice chancellors across the Bharat. First, Professor Girish Chandel, Honorable Vice Chancellor Indira Gandhi Krishi Vishwavidyalaya Raipur, and Professor M. Hanumanta, sir. He is also joined our program. He is a Vice Chancellor of US, Raichur, Karnataka. He is also our partner in organizing this type of esteemed international programs. And we have also Professor Prem Prakash Singh, sir. He is also Vice Chancellor, Sant Gahira Guru Vishwavidyalaya Sarguja, Ambikapur, Chhattisgarh, India. And we have a Director of ICR, Atari, Jabalpur, Madhya Pradesh, Professor S.R.K. Singh. We are welcome you, sir. And Professor Ravi R. Saxena. He is the youngest, most of the Vice Chancellor of Mahatma Gandhi Udyaniki Evam Vaniki Vishwavidyalaya Durg, Chhattisgarh. We welcome you all, sir. And we have also a dignitary like Dr. Sarda Prasad Tripathi. He is a registrar at Sant Gahira Guru Vishwavidyalaya, Sarguja, Ambikapur, Chhattisgarh. And this program is organized and by the Vital Biotechnology and AFTEFS, AFTEFS. Their Secretary President, Mr. Dr. Jitendra Mehtaji. We all welcome you, sir. And we are also welcome our HOD, Engineer Yashwant Kumar Patel, for their presence. And we are also welcome to our Dr. Associates, our Associate Dr. Manoj Kumar Jharia, Assistant Professor and HOD, Vishwavidyalaya. We will all witness transforming era of our profit culture. As we know that our agriculture is the first human revolution technique that is called agriculture. So we are very happy to give a title like this, agriculture and food sector. Just to guys, yes, speaker and participants, our contribution and insight and valuable. We are honored to have you with us, sir. And we have more than 300 participants across all over the world. That is a truly international conference. We are thank you. We are all thank you to all our joint participants, member and our member. Thank you all. You, Dr. Himan, ma'am.
Thank you, sir. Thank you much for providing us providing the very great of this national conference. You have said it rightly to have such activities among us. So moving forward, it's time for the release of the official abstract book or souvenir of this conference. We just have to wait for a few. So, dear participants, on your screen you can see the cover page and of this international conference, and also some of the blessings which we have received from our prestigious institutes and their vice chancellors and other dignitaries. So, after the release of the souvenir of this conference, it's time for a welcome from the chief guest of this conference. So, first of all, yeah. Thank you, sir. So now we are officially done with the release of the conference souvenir. So now it's time for a welcome note from our chief guest of this conference. So first of all, I would like to invite Professor M. Hanumanthappa, sir. Sir is Honorable Vice Chancellor of University of Agriculture Sciences, Raichur, Karnataka. So before Sir take over this podium and enlighten us with his wise words, I would like to tell our participants a little bit about Sir and the contribution he has made to the Indian agriculture and we can say to the global agriculture. So, Dr. Hanumanthappa sir is specialized in the field of agronomy, weed management, soil fertility management, and agroecology systems with MSc and PhD in agronomy. Sir has developed 11 varieties and numerous technologies for the benefit of farming community. Sir has handled more than 100 research projects with total budget out of rupees 725 lakhs as PI funded by RKVY ICR, IMD, Government of India, Government of Karnataka, and also worked as co-PI of more than 12 projects. Sir has been bestowed with numerous prestigious national and international awards, and Sir has a vast experience of 32 years in research, teaching, and extension. Sir has made significant contribution in various in the development of various technologies, and I would like to enlist a few of them. So technologies like contribution in development and popularization of non-herbal tea technology with immense commercial potential and released many technologies for the its package of practices. And Sir has worked for increasing the production of post harvest technology of the products with value addition of for rice, cashew, and help the farmers of coastal districts of Karnataka. Apart from this, Sir has also worked on pineapple, spices, groundnut, and Sir has organized more than 500 field days and 10 Krishi Melas and 200 training programs towards effective dissemination of production and productional technologies of various crops. Sir has over 150 research papers published, 22 books and a book chapter in national and international journal, and also Sir has edited a lot of number of books and 50 popular articles, extension bulletins, and many other publications. So the list of his contribution and his expertise is very long. It's very difficult to enlist all his contributions and specialization in such a short span of time. So without wasting so much of our time, I would like to invite Sir to enlighten us all with his words.
Thank you, Imani Gautam, for your nice introduction. So, first of all,
Thank you, sir.
I congratulate the organizers for organizing the fourth international conference on recent advances in food security, veterinary, and allied sciences for livelihood and environmental sustainability for 3 days conference. So, first of all, Professor Giri Chandal, Honorable Vice Chancellor of Indira Gandhi Krishi Vishwavidyalaya, and Professor Prem Prakash Singh, Honorable Vice Chancellor.
Hello. Sir, इसके बाद directly प्रेम प्रकाश सिंह सर का जाएगा ना?
Okay. Is it? Hello?
Yes, sir.
Is it audible?
Yes, sir.
Audible, sir.
Okay. So, Professor Premchand Prakash, Honorable Vice Chancellor, Sant Guru Vishwavidyalaya, Ambikapur, Chhattisgarh, and Dr. Professor S.R.K. Singh, Director ICR, Atari, Jabalpur, Madhya Pradesh, and many other dignitaries joined here, and many of the young scientists, professors, associate professors, and assistant professors, and also PG students. This in collaboration with many other universities, especially collaboration with the US Raichur, Atal Vishwavidyalaya, Bilaspur, SGGV, Ambikapur, and collaboration with the Agriculture Forum for Technical Education of Farming Society, and Vital Biotech Group, Kota. So with this organization, this conference, 3 days conference has covered like widely areas, especially for the meeting out the sustainable developmental goals. Nearly we have 17 sustainable goals. That out of that, 3 or 4 are very much required with respect to the 6, 7, and 11, 12, 13. 13 is very important for us. What is the fate of the above the ground and below the water? So that what is the life we have to think, and sustainable agriculture. And we have covered almost all the aspects of agriculture and allied sector, especially agriculture, horticulture, animal husbandry, fisheries, forestry. All these areas is very much required in order to maintain the sustainable income as well as sustainable soil sustainability and production sustainability. These resources very effectively utilized with respect to the maintenance of the soil sustainability or sustainable production with respect to the farmer's point of view, especially agriculture combined with animal husbandry. This is very much required because of animal husbandry. Output can be utilized for the input for the agriculture, so that we have to manage the resources very well with respect to the resource conservation itself, so that it requires the very wide areas of the agriculture, especially agriculture, horticulture, animal husbandry, forestry. All these are very much required in order to maintain the soil sustainability and crop production, so that by 2050, we have to increase the production nearly another 70% of the population. So population goes on increasing. We have to meet the increasing demand population to maintain the soil sustainability is very much required, particularly soil carbon. So carbon, we are always losing the soil carbon with different areas, especially in our Karnataka, we are almost 0.3 to 0.5. At least 0.5 is required, 0.5 to 0.75. In coastal district, there is a increasing, mainly because of lot of organic litter, and also they have a lot of greenery, and also we have very good rainfalls. And in dry regions, especially rain-fed area, we are facing a lot of problem with respect to the maintenance of the soil sustainability is concerned. So that we have to use the resources very effectively. In this area, lot of papers has been covered with respect to the biotechnology aspect, microbiological aspect, and as well as agronomy, entomology, pathology, and climate, especially climate change. Presently, we are facing a lot of problems with respect to how we have to deal the climate changes. So either breeding the variety suitable for the climate change, and also agronomic practices, especially management of water, weed, and other aspects. Especially we have different types of weeds that also covered the many of the papers. I gone through the many of the papers. Really, it is a wonderful, and it will be helpful for the young researchers and assistant professors to develop their career for the interaction with the eminent speakers, highly intellectual speakers are there from the India as well as abroad. So other 7-8 countries also joined for this conference. 3 days conference is very useful for the young minds, researchers, how we have to plan for the research that should be sustainable manner, and it should be academia and industries interest. Especially many of our students may not be perform in the industries in such a way that we have to manage our research, and as well as our teaching, it should be useful for the industry, so that they can get the very easily by our students acquire the jobs, especially industry. Based on the industry needs, we have to work. So we have to plan for this such a way that definitely many of the policy makers, industrialists, and researchers and extension workers are joined here. This should be useful for the all the areas, especially it will be useful for the this conference, not just gathering the opportunities for young research to gain the inspiration, expert shares for their wisdom, for their industries, and contest with the ground-breaking advancement and will shape the future global agriculture research requirement. I wish you all the best for everyone for 3 days conferences successfully. It will bring the recommendation useful for the farming community, and it should be technology, viable technology, and good recommendation for the future policy makers and also researchers and industrialists. I thank the organizer giving the opportunity to share with the few words. Thank you, and our university is also one of the co-organizer with respect to this. Our university, we have a good with respect to the AFVAL accredited labs, especially pesticide residue lab, food microbiology lab, and also pesticide formulation testing lab and nanotechnology and food raising and processing. We have a very good with respect to the agriculture and agriculture engineering. Both the colleges we are pioneer in the millet processing and value addition. So that value addition is very much required at present days, but in order to avoid the price crisis, price problem with respect to some of the perishable goods. So that also we have and many startups. So that will also helpful for the farming, so they can get their products value addition, and also many other aspects we can visit. And we have a very good seed of the quality of seeds is very, very important. That also we have to take care, and also solar, we have to harvest the solar energy, and also we have to plan the artificial intelligence. Actually, we include the digitalization in agriculture, particularly artificial intelligence, IoTs, IOF, and also sensor-based irrigation, drones, many other things. We are also working on that drones, how we are getting, and also identify the disease or pest in advance by drones, by image processing techniques. These things definitely we can do with the new young researchers for the next generation. They have to take care of all these things. I thank the organizers giving me the opportunities to share few things. I wish you all the best to everyone, and thank you, one and all.
Thank you, sir. Thank you for your blessings, and thank you for enlightening us with your thoughts. And it is indeed a very proud moment for all of us to share a common platform with you and to interact directly with you. So thank you, sir, and thank you for all your contributions which you have made to the Indian agriculture.
Thank you. Thank you, ma'am.
Thank you, sir. Thank you so much. So participants, moving forwards, it's time for another note from our chief guest, Professor Prem Prakash Singh. Sir is Honorable Vice Chancellor of Sant Gahira Guru Vishwavidyalaya Sarguja Ambikapur, Chhattisgarh. So, Professor Prem Prakash Singh, sir, has pursued his primary higher education from Bihari University examination as direct GRF during 1983 to 84, which was the first time when it was organized in the country. And subsequently, sir got selected in research program of milestone institutions like Bhabha Atomic Research Institute, Vishwabhyalaya Shantiniketan. And his area of research include biochemistry, biophysics, hormone biosynthesis, free radical biochemistry, biochemical toxicology, and environmental physiology. To his credit, sir has published numerous research papers with world's leading publishing houses such as Academic Press, Elsevier, Ville, Springer, Taylor and Francis, CRC Press, and American Association of Experimental Geologists USA, American Association of Immunologists USA, and many more to be mentioned. Sir underwent postdoctoral research from East Tennessee State University, USA, and during research he awarded Student Prize Award along with special recognition. In the field of immunology, sir has co-authored a book with Dr. Vajpayee, who is one of the renowned immunologists of the globe. Dr. Singh is an esteemed academician and has delivered his service as selected member of DST, and sir has also got recognition as distinguished, dedicated, inspiring teacher and scientist by Indian Academy of Science, Bangalore. Along this, sir is a founder of Biotechnology Department in Madhav Institute of Technology and Science, Gwalior, of undivided Madhya Pradesh and Chhattisgarh. This was the first initiative to open this biotechnology course in the engineering domain. Throughout his teaching career, sir has served in the remote and backward scheduled tribe-dominated rural areas of Chhattisgarh. Before joining the present position of Honorable Vice Chancellor, sir was serving in tribal-dominated backward rural areas, Kusmi, for last 8 years. And during his entire tenure of government service at higher education, he emphasized quality education and research and raised his voice against institutional corruption. So, dear participants, it's our privilege to be here and to be able to listen the thoughts of such honorable dignitaries. So, sir, I would like to welcome you and to hand over this virtual podium to you for your welcome note.
Welcome. मैं सबसे पहले सबको प्रणाम करता हूं और इस चतुर्थ अंतर्राष्ट्रीय कॉन्फ्रेंस के अतिथि माननीय कुलपति महोदयों, गेस्ट ऑफ ऑनर महोदय, के नोट स्पीकर्स, स्वागत समिति सदस्यों एवं सभी शोधकर्ताओं छात्राओं को अपनी शुभकामनाएं बधाई देता हूं। कॉन्फ्रेंस के निदेशक आदरणीय डॉक्टर जितेंद्र मेहता जी विशेष धन्यवाद के पात्र हैं, जिन्होंने इस महत्वपूर्ण कॉन्फ्रेंस की अगुवाई की है। मैं संत गहरा विश्वविद्यालय के 2 वरिष्ठ एवं प्रतिभाशाली प्राध्यापक डॉक्टर मनोज कुमार झारिया जी एवं डॉक्टर अनर बनर्जी को भी धन्यवाद देता हूं, जिन्होंने इस कॉन्फ्रेंस में अपनी अग्रणी भागीदारी बनाए हुए है। मैं 3 दिनों तक चलने वाली अंतर्राष्ट्रीय कॉन्फ्रेंस की सफलता के लिए मंगल कामना भी करता हूं। मैं अपने एक लघु व्याख्यान में सी रिएक्टिव प्रोटीन को शामिल किया हूं, जिस पर शोध करने का मुझे अवसर मिला है और यह बायोमेडिकल साइंस शोध में अपना महत्वपूर्ण स्थान बनाए। व्याख्यान के पहले सबसे पहले मैं अपने राष्ट्र के, देश के आन बान शान तिरंगे को सलाम करता हूं और जो मेरा अभी टॉपिक है सी रिएक्टिव प्रोटीन, उसके करेंट ट्रेंड्स एंड फ्यूचर पर्सपेक्टिव पर अपनी बातों को रखूंगा। हम लोग सभी जानते हैं कि खासकर कोरोना के समय सीआरपी टेस्ट कितना महत्वपूर्ण था। उस दौरान सबसे पहले इन्फेक्शन के मॉनिटरिंग के लिए सीआरपी टेस्ट के लिए ही कहा जाता था, ताकि पता चले कि इन्फेक्शन जो है, वायरल इन्फेक्शन कितना गहरा है भी कि नहीं है, तो कितना सीवियर है। और सीआरपी पूरी दुनिया में जाना गया कि ये इन्फ्लेमेशन बायो मार्कर है और जो ग्लोबल है, ये पेंट्रेक्सिन प्रोटीन है जिसमें 5 सब यूनिट्स है। किसी तरह का इन्फेक्शन हो, इंजुरी हो, ट्रॉमा हो, इसका लेवल तुरंत 100 से 1000 गुना बढ़ जाता है। और हैरानी की बात है, फिर तुरंत विदिन 7 डेज ये बेसल लेवल पर आ जाता है। ये अभी भी मिस्टर से कि आखिर इतना लेवल बढ़ता क्यों, इसका रोल क्या है, चर्चा करेंगे। और डॉक्टर भी अब सलाह देते हैं कि अगर आपको लगातार फीवर है, ठंडक लग रही, चिल्स है, रैपिड ब्लीडिंग है, इरेगुलर हार्ट रेट है, तो हार्ट प्रॉब्लम, ये सब सिचुएशंस में जो सामान्यतः कई और डिजीज सिचुएशंस में आते हैं, एथेरोस्क्लेरोसिस हो, ओबेसिटी हो, टाइप 2 डायबिटीज मेलिटस हो, हार्ट डिजीज हो, खासकर इन्फार्क्शन, एलर्जिक रिएक्शंस हो, सेप्सिस हो, कैंसर हो और इन्फेक्शंस, वो सलाह देते हैं कि आप सीआरपी टेस्ट जरूर करवाएं, ताकि जाना जा सके कि स्थिति क्या है आपके शरीर में, इन्फ्लेमेशन का और तमाम डिजीज सिचुएशन। सीआरपी रिसर्च के दौरान इसमें सीआरपी मॉलिक्यूल्स आप देख सकते हैं, जो टिश्यू पर भी जो इलेक्ट्रोमाइक्रोग्राफ है, जब टिश्यू पर डिपॉजिटेड है 5 सब यूनिट्स। और उस तरह मैं शेयर करना चाहूँगा, मुझे इसी कार्य के लिए सीआरपी रिसर्च के दौरान हार्वर्ड विश्वविद्यालय में जाने का मौका मिला था और वहाँ भी अच्छे इंटरेक्शंस हुए। और इसी दौरान मैं जब एक सीआरपी लेक्चर के प्रेजेंटेशन में था, जो रिसर्च पर प्रेजेंटेशन में था, मुझे बड़ा खुशी है कहने में कि जो हमारे फ्रांसिस क्रिक थे, उनके पोस्ट डॉक्टरेट बुढ़ापे की उम्र में भी वो भी प्रेजेंटेशन दे रहे थे, जिन्होंने पॉलीराइबोसोम का डिस्कवरी किया है। तो मैं प्रभावित हुआ इनके इस तरह के उत्साह से कि इस उम्र में भी शोध कर रहे हैं, उसको प्रेजेंट करने के लिए बहुत दूर से वो बॉस्टन आए हुए थे। नेक्स्ट, फ्रांसिस की जिनकी बात कर रहा था, वाटसन के साथ डीएनए मॉडल जो विकसित किया वो दिखाई दे रहे हैं। नेक्स्ट, ये सीआरपी की बात हम जब शुरू करें फिर से लौटना होगा उसके डिस्कवरी पर। प्रोफेसर ऑसवर्ड थियोडोर एवेरी, जिन्होंने डीएनए को, जो ग्रिफिथ का एक्सपेरिमेंट सब लोग जानते थे कि एक हेरिडिटरी प्रिंसिपल है जो ट्रांसफर होता है, स्ट्रेप्टोकॉकस बैक्टीरिया के एक्सपेरिमेंट के दौरान पता चला जनरेशन। लेकिन उस समय ज्यादा इस बात की चर्चा थी कि प्रोटीन जेनेटिक मटेरियल है, हेरिडिटरी फैक्टर है ग्रिफिथ का। थियोडोर एवेरी ने प्रूव किया निमोकॉकस निमोनिया पर। ग्रिफिथ का काम था स्ट्रेप्टोकॉकस निमोनिया पर। इन्होंने बताया कि प्रोटीन नहीं डीएनए, और इन्हें नोबेल प्राइज के लिए काम हो रहे थे। ग्रिफिथ का जो बाइंड करता है के सेल वॉल से और फिर प्रेसिपिटेट के रूप में आता है, और उस प्रेसिपिटेशन के दौरान जो उनका आइसोलेशन फ्रैक्शन था, उसके सी फ्रैक्शन जो उन्होंने नाम दिया था, उसी में इस प्रोटीन का अंदेशा लगा और इसका नाम दिया गया सी रिएक्टिव प्रोटीन, जिसका स्ट्रक्चर बाद में जो है मैकलिन मैकार्टी जो है, जो डीएनए जेनेटिक मटेरियल के बारे में बताए थे, उन्होंने इसका स्ट्रक्चर पता लगाया, उसकी विशेष प्रोटीन जानकारी उन्होंने आगे बढ़ाया बात को, और सी एक्टिव प्रोटीन पर रिसर्च जो है बहुत तेजी से आगे बढ़ा। जैसा हमने कहा कि इन्फ्लेमेशन बायोमार्कर है, और इन्फ्लेमेशन एट द रूट ऑफ मोस्ट डिजीजेस है, इवन कैंसर, कोलिटिस, हेपेटाइटिस, आपका मेनिंजाइटिस, तमाम तरह के रिमेटिक फीवर्स, इन सब में देखा गया कि इन्फ्लेमेशन, इन्फेक्शन तो माना ही जाता है, इन्फ्लेमेशन रूट कॉज है और वो रहता ही है। उससे फिर स्थितियाँ जो है ज़्यादा सीवियर हो जाती है। ऐसे इन्फ्लेमेशन को माना जाता है कि डिफेंस मैकेनिज्म है, जो बैक्टीरियल इन्फेक्शन को जो है प्रोपेगेशन को, मल्टीप्लिसिटी को इनहिबिट भी करता है। तो बायोमार्कर के रूप में सीआरपी का इन्फ्लेमेशन का उभरना इसलिए भी महत्वपूर्ण हो जाता है कि क्लिनिकल एप्लीकेशन इसका शुरू हो गया है, और इसका ये ड्रग थेरेपी भी विकसित किया जा रहा है। आगे हम चर्चा करेंगे। इतने सारे जो डिजीज है, ओबेसिटी, डायबिटीज, तमाम डिजीज आप जो देख रहे हैं हमारे प्रेजेंटेशन के स्लाइड में, वहाँ इन्फ्लेमेशन है। और नेक्स्ट नेक्स्ट नेक्स्ट, और इन्फ्लेमेशन का आप जानते हैं, हीट, रेडनेस, स्वेलिंग, पेन, लॉस ऑफ फंक्शन, खास लक्षण होते हैं, खास लक्षण होते हैं। और कुछ स्लाइड्स आप देख सकते हैं, तमाम तरह के इन्फ्लेमेशन के ये जो है पैटर्न है, जो मॉर्फोलॉजिकली दिख रहा है, क्लिनिकल मैनिफेस्टेशन में ये सब आते हैं। तमाम तरह के नेक्स्ट एक्ने जो होते हैं, जब सीवियर हो जाता है, आप देख सकते हैं ये सब इन्फ्लेमेशन दिख रहा है आपको। और बहुत कम जो है हम लोग स्लाइड ले रहे हैं, बहुत दिखाया जा सकता है। और ये जो है रिमेटॉइड आर्थराइटिस में जो है जॉइंट्स जो आपके हैं वहाँ जो है इन्फ्लेमेशन यही बातें होती हैं। और सीआरपी को अगर हम देखें कि जो है बायोमार्कर इन्फ्लेमेशन है, जो बेसल लेवल इसका अगर 0 के आसपास रखें, जनरली 0.8 मिलीग्राम पर लीटर होता है और मान के चलिए इनिशियल है और उसको 0 के आसपास रखें, जो रेड लाइन देख रहे हैं, तो तमाम प्लाज्मा प्रोफाइल जो है प्रोटीन्स के, कई प्रोटीन्स प्लाज्मा में है, सीआरपी 100 से 1000 टाइम्स बढ़ जाता है और विद इन 7 डेज इसका लेवल पे नीचे आता है और बेसल लेवल को प्राप्त करता है। सीआरपी के साथ साथ कुछ और प्रोटीन्स हैं इन्फेक्शन, इंजरी, ट्रॉमा के दौरान सीरम हेमोग्लोबिन, हेप्टाग्लोबिन, फाइब्रिनोजन, उनका भी लेवल बढ़ता है। ये सबसे सीआरपी सबसे ज्यादा प्रोमिनेंट इसमें है। और कुछ प्रोटीन का लेवल घटता भी है। तो जो एक्यूट सिचुएशन होता है इन्फेक्शन इंजरी का, एक्यूट सिचुएशन बनता है बॉडी के होम्योस्टेसिस का, होम्योस्टेसिस के लिए उसमें जो है कुछ प्रोटीन घटता है, कुछ प्रोटीन बढ़ता है। तो एक एमिनो एसिड पूल से दो तरह की डाइवर्सिटी डाइकोटोमी आती है, जिसका लेवल घटता है उसके कॉस्ट पर किसी का लेवल बढ़ा जाता है। इसको हम लोग पॉजिटिव एक्यूट फिश प्रोटीन्स कहते हैं, जिसका लेवल बढ़े हुए हैं, जिसके लेवल्स घटे हुए हैं, खासकर एल्बुमिन, कॉम्प्लीमेंट C3, ट्रांसफेरिन। तो दो तरह के एक्यूट फिश प्रोटीन्स हुए, जो पॉजिटिव और निगेटिव। और अगर हम देखें कि सीआरपी का लेवल नापे, मेजर करें, और एल्बुमिन का जिसका का बढ़ रहा है का घट रहा है, अगर इसको हम लोग तुलनात्मक इसके लेवल को लें तो, और इसको लिया जाता है सीआरपी एल्बुमिन, और ये परफेक्शन को बात जाती है कि है, इन्फेक्शन है, इन्फ्लेमेशन है, और इन्फ्लेमेशन का स्टेटस क्या है। नेक्स्ट, ये आप देख सकते हैं, वही बात है कि पीक 72 आवर्स में पीक लेवल सीआरपी का मिलता है। इन्फ्लेमेशन की सिचुएशन या जिस बातों से आए, और वो फिर घटना शुरू हो जाता है 7 डेज। लेकिन जब सर्जरी होती है, ट्रॉमेटिक सिचुएशन आता है और हीलिंग तक इन्फ्लेमेशन की स्थिति बनी रहती है, तो जो परसिस्टेंट लेवल जो बढ़ा हुआ है वो परसिस्टेंट हो जाता है और कुछ और दिनों तक वो लेवल बढ़ा रहता है। नेक्स्ट, अगर आप प्रोफाइल देखेंगे, प्लाज्मा प्रोटीन प्रोफाइल, तो एल्बुमिन फ्रैक्शन है, लो मोलेक्युलर वेट, हाई मोलेक्युलर की तरफ जब बढ़ेंगे, ग्लोबुलिन फ्रैक्शन की से अल्फा बीटा गामा की तरफ जाएंगे, गामा में एंटीबॉडीज मिलेंगे आपको। तो हम देख रहे हैं कि ग्लोबुलिन फ्रैक्शन में ही एंटीबॉडी के आसपास सीआरपी का प्रोटीन का जो है सिग्नल आता है वापस। हाई मोलेक्युलर प्रोटीन है, पेंटामेरिक हम जानते ही हैं, और ये मल्टी फंक्शनल इनेट पोस्ट डिफेंस है। जन्म से ही ये प्रोटीन हमारे शरीर में रहता है और डिफेंस प्रोटीन रहता है। और ये नॉन स्पेसिफिक मार्कर इन्फेक्शन, तो किसी भी तरह का इन्फेक्शन इंजरी हो, नॉन स्पेसिफिकली ये इसको बढ़ेगा। इसलिए बहुत इंपॉर्टेंट ये क्लिनिकल प्रोटीन बन गया है, इसकी मेजरमेंट बहुत जो है पॉपुलर हो गया है क्लिनिकल एस्पेक्ट पर। और ये डिफेंस प्रोटीन इस अर्थ में भी है कि ये बैक्टीरियल जो सेल वॉल है उस पर जो एक बायोकेमिकल पैटर्न है, लैंडस्केप है, उसको बाइंड करता है। बाइंड करने को इसका मतलब ये पैटर्न रिकॉग्निशन मॉलिक्यूल है जो सर्कुलेटिंग फेज में है। पैटर्न रिकॉग्निशन मॉलिक्यूल जो ह्यूमरल भी होता है और सेलुलर भी होता है, ये ह्यूमरल है। और जब ये बाइंड करेगा और कॉम्प्लेक्स बनेगा सीआरपी और बैक्टीरियल सेल वॉल कंपोनेंट, खासकर जो उसमें हम पहले से जानते हैं, इस सेल वॉल में अगर जो है हमारा सी पॉलीसेक्राइड, इसको देखेंगे आज, इसे बाइंड कर, तो फिर ये रिकॉग्नाइज हो जाता है मैक्रोफेज मोनोसाइट से, और फिर ये फेगोसाइटाइज हो जाता है। इसलिए बैक्टीरियल क्लीयरेंस में सीआरपी का बहुत बड़ा रोल है। और ऐसे मॉलिक्यूल जो बैक्टीरियल फेगोसिटोसिस को बढ़ाए, उस पर बाइंड करके इंफॉर्मेशन दिलवाए मोनोसाइट और मैक्रोफेज को, और फिर उसको हटाया जा सके सिस्टम से, उसको हम लोग ऑप्सोनिन कहते हैं। तो नेचुरली सीआरपी भी ऑप्सोनिन कैटेगरी में आया, जिसमें कॉम्प्लीमेंट भी आते हैं, आईजीजी भी आते हैं, और कुछ लेक्टिन बाइंडिंग मैनोज भी सर्कुलेटिंग प्रोटीन्स आते हैं। नेक्स्ट, अब देखें ये सेल वॉल है, और सेल वॉल के ऊपरी लेयर में हम दिखा रहे हैं कि क्वैक एसिड, जो एक्चुअली मॉडिफाइड पॉलीसेप्टाइड है, जिस पर फॉस्फोकोलाइन भी अटैच्ड है, और वही बाइंड करता है सीआरपी। इसका मतलब सीआरपी फॉस्फोकोलाइन से बाइंड करता है, और वो कहाँ है, वो बैक्टीरिया के सेल वॉल पर है, हमारे सेल मेम्ब्रेन में भी है, जो लिपोप्रोटीन्स हैं उस वहाँ भी हैं। इसकी बाइंडिंग लिगेंड जो है वो बहुत जगह फैले हुए हैं, कैंसर सेल में, नॉर्मल सेल में भी, और डैमेज सेल में भी, एपोप्टोटिक सेल में भी, वहाँ आपको फॉस्फोकोलिन है, इंक्लूडिंग बैक्टीरिया। तो जहां जहां फॉस्फोकोलिन है सीआरपी वहां बाइंड करेगी। और उसके अलग अलग रूल्स हैं। नेक्स्ट, ये निमोकोकल पॉलीसेकेराइड है, मॉडिफाइड पॉलीसेकेराइड है, आप देख सकते हैं, और उसी में फास्फोकोलिन है और उस पर सीआरपी बाइंड कर रहा है, और ये बाइंडिंग कैल्शियम डिपेंडेंट। तो सीआरपी के सब यूनिट्स में ऑलरेडी वहां कैल्शियम बंधे रहते हैं, और जहां कहीं फास्फोकोलिन मिला इसको सेक्रेशन के दौरान, सेक्रेशन के बाद ये वहां बाइंड कर जाएगा, चाहे बैक्टीरियल सेल वॉल हो, या आपका डैमेज सेल हो, एपोप्टोटिक सेल हो, कैंसर सेल हो, ये बहुत सारी पॉसिबिलिटीज हैं, जो ऑक्सीडाइज लिपोप्रोटीन हो, जहाँ एक्सपोज हो जाते हैं फॉस्फोकोलिन, फॉस्फोलिपिड बायोमेम्ब्रेन में रहते ही हैं, वो बाइंड करता है। नेक्स्ट, ये वही चीज है, बैक्टीरियल सेल वॉल पर बाइंडिंग साइट्स हैं सीआरपी का। नेक्स्ट, सीआरपी ऑप्शन इन हम लोग चर्चा की, ये आईजीजी कॉम्प्लीमेंट के साथ जो है उस कंपोनेंट कई सारे फैक्टर्स हैं जो बाइंड करते हैं, उसमें सीआरपी भी है, और जब ये बाइंड करते हैं मैक्रोलोज के किलिंग यूट्रोफिल्स टिश्यू मैक्रोस्फीयर जला वो बढ़ा देते हैं, और से जो है इन्फेक्टिव माइक्रो ऑर्गेनिज्म का और इवन डैमेज सेल्स का, कैंसर सेल्स का क्लियरेंस बढ़ जाता है। ये डिफेंस मैकेनिज्म आ गया कई तरह से, ऑप्शन इन है। वही बातें हैं कि जब कोई इन्फेक्शन इंजरी हुआ जो है, तो इननेट तौर पर सीआरपी क्लियरेंस तो कर ही रहा है, और उसके बाद जो है हमारे एडप्टिव इम्यूनिटी से हम एंटीबॉडी प्राप्त करते हैं, और जो है एंटीजन से बाइंड करके जहाँ भी रेसर्कुलेटिंग रहे या इन्फेक्टिव एजेंट के सरफेस पर रहे, एक इम्यून कॉम्प्लेक्स बनाता है, और उससे जो है इंटरल्यूकिन सेक्रेशन शुरू हो जाता है उस एरिया से, वहाँ के टिश्यू से। इंटरल्यूकिन जो लीवर के पास आके, खासकर इंटरल्यूकिन 6, IL-6, IL-1 बीटा लीवर से प्रोड्यूस करता है, सीआरपी सर्कुलेशन में आता है, फिर ऑप्सोनिन की तरह व्यवहार करके कॉम्प्लीमेंट क्लासिफिकेशन पार्ट को एक्टिवेट करता है, और बैक्टीरिया का सेल का लाइसिस भी होता है, ऑप्सोनाइजेशन भी होता है। नेक्स्ट, तो वही बातें हैं, ये पेंटामर है सीआरपी, और बगल में आप देख रहे होंगे इसका 1 सब यूनिट है, जिसमें बीटा चेन का प्रिवलेंस बहुत ज्यादा है। वो ऐसे ही देख रहे होंगे, फॉस्फोकोलिन बाइंडिंग साइट है, 2 कैल्शियम बाइंडिंग साइट है, और इस तरह से हर एक सब यूनिट पर 2-2 कैल्शियम पहले से बंधे रहते हैं, और जहां कहीं भी फॉस्फोकोलिन मिलेगा, जहां कहीं भी वहां वो बाइंड करेगा। नेक्स्ट, ये मैं जो बुक चैप्टर लिखा था, बहुत मुझे बड़ा अच्छा लग रहा है कहने में, लाइफ टाइम एक्सपीरियंस है।
Sir, excuse me, sir.
Yes,
Yes, sir. Yes, sir. Actually, we have a very limited time, and our next guest, he has some important assignment, and his time.
Have some few minutes.
Yeah, we just want you to conclude this in hurry. Yeah.
Actually, I had the idea that I'll give some time. Okay, okay.
Yeah, we have limitation of time.
Yes, okay, next. Now I can say कार्डियोवास्कुलर डिजीज में इसकी मॉनिटरिंग बहुत इंपॉर्टेंट मानी गई है। लो रिस्क पेशेंट अगर 1 मिलीग्राम से कम है हमारा पर लीटर सीआरपी लेवल, लो रिस्क है। 1 to 3 मिलीग्राम में मॉडरेट रिस्क है, 3 मिलीग्राम से ज्यादा 10 तो हाई रिस्क है। लेकिन जब एथलेटिक प्लेट का पंक्चर होता है वो तो हार्ट अटैक के समय तो 10 मिलीग्राम पर लीटर भी चली जाती है। आप देख सकते हैं एथलेटिक प्लेट साइट पर सीआरपी डिपोजिशन बहुत ज्यादा है पेंटामिल के रूप में, और कम में कम है, सीवियर होने पर ज्यादा है। तो सीवीडी में कार्डियोवास्कुलर डिजीज में इसका इन्वॉल्वमेंट देखा गया, डिपॉजिट देखा गया। नेक्स्ट, लेकिन क्यों है वहां हमको पता नहीं। यहाँ देखिए एथलेटिक प्लेट है और बहुत सारा सीआरपी मॉलिक्यूल्स है यहाँ, लेकिन क्यों है ये हमें पता नहीं। तो इसे कम करने के लिए साइट पर जहां भी रैट मॉडल में देखा गया। नेक्स्ट चलो, और नेक्स्ट चलो, शायद 2000 चलो, और चलो, और चलो। हाँ, रैट मॉडल में देखा गया कि अगर एक कंपाउंड बनाया गया, सिंथेसाइज किया गया, जिसमें फॉस्फोकोलिन दोनों एंड पर था, बीच में हेक्सेन था, उसको कहते हैं 1,6 फॉस्फोकोलाइन हेक्सेन। अगर उसको सर्कुलेशन में उसके सीआरपी तो है ऑलरेडी एस्ट्रोस्कोपिक प्लेट पर, अगर ये कंपाउंड इंजेक्ट करते हैं, ये 2 पेंटामिल को बाइंड करता है, जो सीआरपी बाइंड करना था टिश्यू साइट पर प्लेट के, अब ये बाइंड कर रहा है सेक्शन से, अब वहां नॉन फंक्शनल हो गया। ये पाया गया रैट मॉडल में कि सीआरपी एज ए ड्रग इंट्रोड्यूस किया, एज ए ड्रग हेक्सेन को जो बाय फॉस्फोक्लाइन हेक्सेन था उसको इंट्रोड्यूस करें तो हमारा इन्फेक्शन साइज कम हो जाएगा, बिकॉज उससे सीआरपी को इंगेज कर लेगा साइट पर और हमको टिश्यू डैमेज कम होगा। जो एक और बात मैं बता, और ये वही चीजें हैं दो पेंटामर को इंगेज किए हुआ है, ये 1,6-5 फॉस्फोक्लाइन हेक्सेन है। अब ये पेंटामर दो पेंटामर को इंगेज कर लिया, अब ये दो पेंटामर जो था, जो फ्री था, अब डैमेजिंग साइट पर बाइंड नहीं करेगा और इन्फेक्शन कंट्रोल जो है कंट्रोल में आएगा। एक बात जो मैं कहना चाहूंगा फाइनली, तो ऊपर चलो ऊपर चलो, नेक्स्ट 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 नेक्स्ट, हाँ यही जो इसका लेवल तत्काल बढ़ जाता है बहुत एक्यूट इन्फेक्शन के बाद सिचुएशंस में, इंजरी में, तो ऐसा इसी वजह से होता है। ये प्रमोटर है सीआरपी का। तो मैंने ये पाया कि जो इसका प्रमोटर है +1 से आरएनए बनेगा इसका, फिर जो बाद में ट्रांसलेट होकर सीआरपी प्रोटीन सबमिट बनाएगा। ये बाइंडिंग साइट्स के प्रमोटर पर ट्रांसक्रिप्शन फैक्टर्स का जो आरएनए पॉलीमरेज की प्रोग्रेशन को घटाता है या बढ़ाता है ट्रांसक्रिप्शन के दौरान। जब लेवल बेसल है तो ये देखा गया कि प्रमोटर साइट पर सीबीपी जीटा आरबीपी जेके के साथ बाइंड करके उसके लेवल को घटाता है ट्रांसक्रिप्शन के। लेकिन जब तो ये जो जोड़ा है सीबीपी बीटा आरबीपी जेके ट्रांसफर फैक्टर का रिप्लेस हो जाएगा सीबीपी बीटा से और एनएफ का भी प्रोटीन P50 से। तो ये जो स्विच है मॉलिक्यूलर जेनेटिक मॉलिक्यूलर स्विच, यही इंपॉर्टेंट है कि सीआरपी जो है एज ए बायोमार्कर स्टंट राइज का लेवल का क्यों होता है, इसको ये शोध मेरे जनरल ऑफ न्यूरोलॉजी में पब्लिश है और ये एक महत्वपूर्ण रहस्य जो सुलझा। और इसके बाद जो सीआरपी की स्थिति है, इसको अब एज ए ड्रग इसको रिवाइव करने की कोशिश की जा रही है और उसके लिए इसका कई म्यूटेंट वर्जन है, कई स्मॉल मॉलिक्यूल्स पैदा किया जा रहा है, बनाया जा रहा है जो सीआरपी को के बाइंडिंग को, इसके डैमेजिंग प्रॉपर्टी को इंटरफेयर करे और इस दिशा में कार्य आगे हो रहा है। फ्यूचर में ट्रांसफर करना जरूरी है दिशा में कि साइट पर पेंटामर जो सीआरपी मोनोमर में बदल जाते हैं, एक उनके बीच में एक बैलेंस जो है इक्विलिब्रियम की बात रहती है। दूसरी बात, अभी तक हम ये नहीं जान पाए हैं कि सीआरपी का लेवल तुरंत इतनी ज्यादा क्यों बढ़ जाती है अन्य प्रोटीन की तुलना में। तो फ्यूचर में इस डायरेक्शन में मैकेनिज्म तो पता चल गया, उसमें मेरा भी काम है, लेकिन ऐसा होता क्यों है ये फ्यूचर पर्सपेक्टिव की बात है, आगे की बात है। बहुत बहुत धन्यवाद, थोड़ा सा ज्यादा समय लिया और मैं फिर से आपको बधाई देता हूँ, शुभकामनाएं देता हूँ कि 3 दिन तक चलने वाला ये इंटरनेशनल कॉन्फ्रेंस सफल हो और सबों को बहुत लाभ हो और साइंस की दुनिया में ये इस तरह की जो बड़े वाइड कॉन्फ्रेंस कवरेज है आगे जाता रहे। बहुत बहुत प्रणाम, सबको धन्यवाद।
Pranam sir, thank you and thank you for sharing your experience and expertise with us. We have limited time, so that's why we have
ठीक है,
to conclude your
बहुत
talk.
बहुत धन्यवाद आपका।
Otherwise we
मानी
like
गौतम
so
जी,
much.
बहुत बहुत धन्यवाद आपका।
Yes sir, thank you sir, thank you so much. Thank you for taking out your precious time and enlightening us all with all your experience. So, dear participants, this is now this is time to for address from our next guest of honor of this conference. So, the guest of honor of this conference, Professor SRK Singh, sir. So, sir is director of ICR Atari, Jabalpur, Madhya Pradesh. So, sir has obtained bachelor degree in agriculture from Institute of Agriculture Sciences, Banaras Hindu University. Sir has completed his MSc and PhD in Dairy Extension at National Dairy Research Institute, Karnal. Sir has received merit scholarship during his entire academic period, and during 1999, Sir has topped ARS examination and started his career as scientist of agricultural extension at ICR Vivekanand Parvatiya Krishi Anusandhan Sansthan, Almora, Uttarakhand. Sir is recipient of numerous awards, including SEE Fellow Award, ISEE Fellow Award, Best Mobilizer Award, Best Extension Scientist Award, Best Extension Professional Award, Young Scientist Award, Agriculture Today Award, along with Dr. Bashant Rao Nayak Award for Outstanding Research and Application in Dryland Farming System 2021 from ICAR. Sir has numerous publications, including 130 research articles in various national and international reputed journals, 18 books, 53 bulletins, 16 book chapters, and many more. Sir is presently engaged in research and extension with KVK system of ICR in Central and Eastern India. Sir has been instrumental in initiating and implementing concepts like KVK KMA, KVK Ring, KVK Technology Park, KVK Atma Convergence Model, methodological initiatives for extension research, Nutri-Smart Village, agripreneurship, etc. Sir has been member of various committees of ICR and state agriculture universities. So the contribution of Dr. SRK Singhs are endless, and enlisting all of them in such a short span of time is not possible at this moment. So with all these words, I would like to invite Dr. SRK Singh to address us all and to share his thoughts with us at this virtual platform. So over to you, sir. So dear participants, sir has some important assignments to attend. So sir will be joining us in a while. So till then we are inviting our next guest of honor, Professor Ravi S. Saxena, sir. Sir is Honorable Vice Chancellor of Mahatma Gandhi Udyaniki एवं वानिकी विश्वविद्यालय दुर्ग छत्तीसगढ़. Sir is a renowned academician with over 30 years of experience in higher education and agriculture innovation. Sir has guided number of students, many of whom now hold prestigious global positions. Sir is recognized nationally and internationally, and sir has received Normal Borlaug Fellowship in 2007 from USA and International Fellowship from International Rice Research Institute, Philippines in 2001. Dr. Saxena has made impactful contribution to e-governance, earning the National E-Governance Gold Award in 2023 for AI-driven smart farming and the Silver Award in 2016 for MIS development in agriculture. Sir has completed leadership training through the LEAP program at Monash University and AMU, enhancing his skill in university governance and research. Currently, sir collaborates with IIT Bhilai to digitally transform agriculture for tribal farmers. Sir has successfully led multiple government-supported research projects and marking him as a key figure in agriculture education and digital innovation. So in this digital world where we are all connected digitally, so we have Professor Ravi Saxena, sir, with us, who is working very hard to make it possible for agriculture to go digital, for farmers to go digital, and we are very lucky today that we have him with us. And so with these words, I would like to invite Professor Ravi Saxena, sir, to enlighten us all with his thoughts. Over to you, sir.
Thank you, madam. Thank you so much for giving me the introduction. Very good afternoon to all of you, Honorable Vice Chancellors, Directors, respected organizer, distinguished guests, esteemed delegates, faculty members, and dear students. It is indeed a privilege and great honor for me to be here as a guest of honor at the prestigious International Conference on Recent Advances in Agriculture, Food Security, Allied Sciences for Livelihood and Environmental Sustainability. Agriculture today is not just about the producing more. It is about the producing smarter, safer, and manner that respect both nature and the people. So, whether it is use of artificial intelligence, blockchain technology, IoT in precision farming or conservation of biodiversity or promoting climate resilient crops, the role of research, collaboration, and policy alignment cannot be overstated. I am happy to see that this conference bringing together experts from diverse fields, agriculture, environmental science, food technology, social sciences, to share the ideas, innovations, and experiences that can guide our path forward. The presence of young researchers and students here gives me the hope for the agriculture and sustainability in the hands. I would like to extend my heartfelt congratulations to organizing committee for curating such a relevant and dynamic platform. I am confident that the outcome of the conference will contribute meaningfully to positive policies, practices, and partnerships that shape a more sustainable future for all. Thank you once again for this honor. I wish the conference grand success and look forward the deliberation ahead. Jai Hind, Jai Kisan. Thank you so much.
Thank you, sir. Thank you for your blessings and thank you for your good wishes for the conference. And we are truly so honored to have you on board today. So thank you for taking out your precious time. So the participants, now it's time to move forward. So with these words, I would like to invite our next guest of honor, Dr. Sharda Prasad Tripathi, sir. Sir is Registrar of Sant Gahira Guru Vishwavidyalaya, Sarguja, Ambikapur, Chhattisgarh. So sir has pursued his postgraduate and PhD work from Hari Singh Gaur Central University, Sagar, Madhya Pradesh in sociology. Further, sir has secured MPhil degree from Barkatullah University, Bhopal, Madhya Pradesh, and his thrust area of research is based on tribal communities of Bharia tribes of Patalkot area of Madhya Pradesh. To his credit, sir has also associated in World Bank funded projects on rural development. He is also a dynamic person having wide expertise in working in various reputed institutions in different capacities. He has also published various research papers, articles, reviews in reputed journals and magazines. To his credit, he has 30 years of academic and administrative experience at college and now presently serving as a registrar of Sant Gahira Guru Vishwavidyalaya Sarguja Ambikapur. As a researcher and as a student of sociology, he is actively involved in various forms of social work, rural development, human development, and tribal welfare apart from research and development activities at university level. So with these words, I would like to invite sir to address us all. So over to you, sir. Dear participants, we have to wait for few moments as sir is facing some technical issue. So we are waiting for him so that we can proceed further. I would like to request you all to wait for a while. Thank you. Thank you for patiently waiting, participants. So I would like to inform you all that due to some technical issue, sir is not able to join us at this moment. And without wasting much time, we are moving forward. And with moving forward, I would like to invite uh, President of Agriculture Forum for Technical Education of Farming Society Kota, the President of this society, Dr. Jitendra Mehta, sir, for his address to all of us. So I would like to hand over this virtual podium to Dr. Mehta.
Thank you, Dr.
Over to
Himani.
you, sir.
Thank you, Dr. Himani, to provide me this opportunity to express of my words in front of wonderful audience. Many people have made extraordinary contribution in the successful conduction of 4th International Conference on recent advances in agriculture, food security, veterinary, and allied sciences for livelihood and environmental sustainability. I feel very happy to thank them. And in this series, Dr. Jitendra Mehta, President of Agriculture Forum for Technical Farming Society, first thankful to the chief guest, Professor A.D.N. Vajpayee, sir, Honorable Vice Chancellor, Atal Bihari Vajpayee Vishwavidyalaya, Bilaspur, Chhattisgarh, under whose leadership this organization is working. Sir, your words are always blessing for us. And I thank the Professor M. Hanumantappa, sir, Vice Chancellor, University of Agriculture Sciences, Raichur, Karnataka, and Professor Prem Prakash Singh, sir, Honorable Vice Chancellor, Sant Gahira Vishwavidyalaya, Ambikapur, Chhattisgarh, and esteemed guest of honors, Dr. S.R.K. Singh, sir, Director of ICR Atari, Jabalpur, Madhya Pradesh, Professor Ravi Saxena, sir, Honorable Vice Chancellor, महात्मा गांधी उद्यानिकी एवं वानिकी विश्वविद्यालय दुर्ग, छत्तीसगढ़, and Dr. Sharda Prasad Tripathi, sir, Registrar, Sant Gahira Vishwavidyalaya, Sarguja, Ambikapur, Chhattisgarh, whose guidance we get every step, through we have able to reach this position today. And we hope that we will continue to get your guidance in future also. I express my gratitude to the organizing director, Dr. M.K. Meena, sir, and engineer Yashwant Kumar Patel, sir, who provided unique support in channelizing the entire program. I am grateful from the bottom of my heart to all the organizing secretaries, Dr. Soumitra Tiwari, sir, and Dr. Y.S. Amresh, sir, and Ms. Jaya Mehta, and of course, convener of this program, Dr. Manoj Kumar Jharia, and Dr. Anna Banerjee, whose point-to-point guidance we were able to easily overcome all the difficulties in this program. I am grateful to all the members of National and International Advisory Committee, Advisory Secretary, and Organizing Committee who fully supported us in making this program successful. And I am grateful to all the highly knowledgeable speakers of this conference who, on the basis of their grassroots knowledge, helped in improving the knowledge of all our participants. And on the behalf of AFTEFS and Vital Biotech, I thank our host organization like Atal Bihari Vajpayee Vishwavidyalaya, Bilaspur, Chhattisgarh, University of Agriculture Sciences, Raichur, Karnataka, and Sant Gahira Vishwavidyalaya, Sarguja, Ambikapur, for providing us the opportunity to organize this conference. I hope that we must have lived up in the expectations, and we will continue to get the opportunity to be involved with you in future also. Thank you all.
Thank you, sir. Thank you, sir. Thank you for addressing all of us. And now I would like to invite convener of this international conference, Dr. Manoj Kumar Jharia, sir, for vote of thanks. Sir is assistant professor and HOD at Sant Gahira Vishwavidyalaya, Sarguja, Ambikapur. So over to you, sir.
Thank you very much, Dr. Himani Gautam. First of all, I would like to wish all the participants very good afternoon to all of you, and I welcome you all in this gracious for your gracious present in this 4th international conference on recent advances in agriculture, food security, veterinary, and allied sciences for livelihood and environmental sustainability. First of all, I would like to thank all the collaborating institutions, Sant Guru Vishwavidyalaya, AFTEFS, Biotech, Atal Bihari Vishwavidyalaya, Bilaspur, Indira Gandhi Vishwavidyalaya, Raichur, and US Dharwad University of Agriculture Sciences, Dharwad, Karnataka. I would like to thanks all the dignitaries present here, especially Professor Arun Divakarnath Vajpayee sir, Honorable Vice Chancellor Sir Atal Bihari Vajpayee sir, Atal Bihari Vajpayee Vishwavidyalaya Bilaspur, Professor Girish Chandel sir, Honorable Vice Chancellor IGKB Raichur, Professor M. Hanumantappa sir, Honorable Vice Chancellor US Raichur, Karnataka, Professor P.P. Singh sir, Honorable Vice Chancellor Sant Guru Vishwavidyalaya, Ambikapur, Chhattisgarh, Professor S.R.K. Singh sir, Director of ICR Atari, Jabalpur, Madhya Pradesh, Professor Ravi R. Saxena sir, Honorable Vice Chancellor Mahatma Gandhi Udyaniki एवं वानिकी विश्वविद्यालय दुर्ग छत्तीसगढ़, Dr. Sarda Prasad Tripathi, Registrar Sant Guru Vishwavidyalaya, and Dr. Jiten Mehta sir, the coordinator of this program, and all the organizing secretaries, convener, co-convener, and who those directly or indirectly make this program towards the grand success. I wish all the best and best wishes for the successful ongoing of this 4th international programs who are joined the program now and in upcoming the technical sessions. So thank you very much all everyone for this wonderful and gracious presence. Thank you very much.
Thank you, sir. Thank you so much for your crisp and brief address to all of us. And now moving forward, I would like to invite the keynote speaker of today's session. So the keynote speaker for today's session is Dr. Ram Sarup Meena, sir. Sir is Associate Professor at Department of Agronomy, Institute of Agricultural Sciences, Banaras Hindu University, Varanasi. So, like most of us, sir also belongs to a farmer's family, and coming from a farmer family, sir has maintained his academic record very well. Sir has secured first division in all the classes with triple net, JRF and SRF from ICR, and RGNF awardee from UGC, Government of India. Sir is a fellow of National Academy of Agricultural Sciences, National Academy of Biological Sciences, and fellow of Society for Rapeseed Mustard Research. And he is also a visiting scientist at IRRI SARC under INSA Fellowship. Sir has been awarded with Raman Research Fellowship by Ministry of Education, Government of India. Sir has been listed in the world's top 2% scientists by the Stanford University, USA, and elsewhere. He is listed in the at the 8th position out of 100 scientists from India in forestry and agriculture sciences by AD Scientific Index at the global level ranking. He has completed his postdoctoral research on soil carbon sequestration under World Food Prize 2020 and Nobel in Contribution 2007 laureate Padma Shri Professor Ratan Lal, distinguished scientist and director at the Professor Ratan Lal Center for Carbon Management and Sequestration Center, the Ohio State University, USA. Dr. Meena is working on soil sustainability, crop productivity, soil carbon stability, carbon credit, regenerative agriculture, circular economy, and resource use efficiency in the current climatic era. Sir has supervised 27 postgraduate and 7 PhD students, and sir has more than 15 years of research and teaching experience at the UG and PG level and also at PhD level. Sir has experience with 10 externally funded projects as PI and co-PI, including the ongoing projects from IOE, BHU, and DST, ICR, Ministry of Education, Government of India. And sir has the right to one patent on low-cost biochar preparation. Sir has numerous publications, including 120 international research and review papers in peer-reviewed reputed journals. And Dr. Meena contributed to the agriculture extension activities in the Honorable Prime Minister of India Villages and working for 3 villages in the mid-forest tribes of Sonbhadra district as an associate coordinator in training meetings, workshops, and farmers' fair. His future target is to reduce the soil organic carbon oxidation and enhance stability in agro-ecosystem for farmers' benefit through carbon credits. So with all this, we all got to know that sir is working very hard in his area of expertise, and we are very excited to for your talk, sir, because we know that today we are going to learn a lot of new things from you. And with these words, I would like to hand over this virtual stage to you. So over to you, sir.
Thank you, Dr. Imani. My voice is okay?
Yes, sir, your voice
Yes,
is
sir.
clear and loud.
Okay, thank you so much. First of all, I would like to say congratulate because just it started of the 4th International Conference from the society side of Dr. Mehta, and so many prestigious dignitaries is here, chief guest and conveners. Like that, Honorable Vice Chancellors, many Vice Chancellors sir, is also there. See, it is pleasure to me. I am also presenting my views in front of the Honorable Chief Guest and Guest of Honors, Professor Vajpayee ji, Professor Chandel ji, Professor M. Hanman Thappa ji, Professor Prem Prakash Singh ji, and Professor S.R.K. Singh ji, Professor Ravi R. Saxena ji, and Professor S.S. Tripathi ji, and Jitendra Mehta ji is also there because he's the organizing man of that one, as well as the convener Dr. M.K. Jharia ji, and my colleague is also, I think, sitting over there, Dr. Banerjee. So it is a great pleasure to me. I have to present my views. So if you will permit, I will share my slide.
Yes, sir, please. Yeah, so kindly switch to full screen mode.
I think I already
Yes,
switched
yes,
it.
sir. Yes, yes, it's
So
visible.
now is okay?
Yeah, sir, now you can proceed.
Okay, thank you so much. Because we all are from the science background. So in the science, every movement is coming something new hypothesis, sometimes we accept the hypothesis and sometimes we reject the hypothesis. As well as a team of scientists always work. No single one can win in the science. As other than this, the many technologies are accepting and rejecting. So according to the time, many technologies are also accepting by the society. So my just as per my career, I'm presenting my own views. So whatever I did my past, on the behalf of that past, I am presenting my views, and my topic is there carbon credit potential in Indian agriculture to regenerate the soil and enhance the farmer income. And whatever the possible policy framework is there, because it is a big word right now, everybody is because suffering from the climate change. And due to the climate change, many things happening at the global level, because climate change, it's, we can say, act locally, think globally. So on this perspective, carbon is the key player in the ecosystem. So my presentation is on the basis of that topic. Yeah. So I will be focused the some part of the green revolution and whatever the impact agrochemical market impacts on the health. Maybe one health mission is also there. So many health is also connected with that. Soil, carbon and climate change, regenerate, because right now we have need to regenerate the soil, and carbon management, carbon credit and stability is more important than whatever we are putting to the soil. And whatever the policy framework is there and how it is linked with our personal life and professional life and food chain. Like that, I'm quoting one example is here. So during the 1950, we are standing to produce the food only 50 million ton. But right now we are standing, it is about 340 and more than 40 million ton. And population is also increasing drastically. And that is the another way it means we are sufficient to feed the population. Even in Corona, agriculture was the one sector was standing with the country. So it is important because we are agriculture based country. So everybody is linked, maybe raw material, everything is coming from the agriculture. So that is the reason agriculture have all solutions. So I will prove how can carbon is important in agriculture and food system. You can see in the figures, continuously we are increasing the nitrogen and many NPK doses, but yield response is decline according to the time. Initially, we got a boom in the production, but right now production system has been declined. So that will be the another issue we can say, but we are totally depend on the fertilizer basis, and in agriculture market we are fertilizer based production is increasing continuously. So it's okay on the basis of the fertilizer, and after the sometime irrigation norms and other these things is there, high yielding variety, it is a joint venture to increase the fertilizer. But in the fertilizer sector right now we are putting lot of subsidy. So it is the matter to, so I think so on the basis of the fertilizer production, we are getting more production, but total amount of the whatever we are putting to the fertilizer sector or how much subsidy is there, it is millions and trillions rupees game. So like that, I am quoting an example, DAP bag. DAP bag price to the farmer is 1,350, and it is vary with the state by state. But actual price is more because lot of amount is going to the private sector. So my point is here, it's okay based on the fertilizer and agrochemical market, lot of subsidies we are getting benefit from the subsidy related to farmer, but trillions of rupees we are wasting to the subsidy. I am not saying so we withdraw the subsidy. My point is here, we think about the how much subsidy it is reliable to give the farmer, whatever the plan to modify at the country level, because it is a big crisis. Like that, here, I think it is in full mood. Yeah. So here we can say example, in the due to the crisis of the fertilizer, farmers were on the road sometime. So even another issue is there, raw material is not available to the fertilizers. So it will be in the near future. Many prediction is there like that a DAP, many fertilizer raw material will be, because we are continuously mining it. So it will be another issue. And another issue with the farmer, over there the crisis, over there the fake. So fake DAP and many type of fake fertilizer is also there. So my point is there fertilizer, farmer is totally depend on the fertilizer, and sometimes they are not getting the good quality of the fertilizer, or even trillions of rupees we are paying to the private sector in terms of the subsidy, and that amount we must be managed. And another issue with the fertilizer and agrochemical market, you can see in the graph, continuously fertilizer graph or consumption is higher and higher side. Parallelly, drug market is going up and up, because it is only because of the we are not getting the good for quality. That's why reason we are crying for the organic farming. It means we are totally go to the, but it is not possible we can fulfill the requirement with the organic farming to the country. But we think about the, we can say integrated approach, something we can cut down from that side, and something we think about the to go to the soil. So that's why reason, if the soil will be perfect, and then you can see the future prediction is there. We need to more $1 million to pharmaceutical industry. It is because of the we are not getting good diet. Whatever we are taking, the plant is taking from the soil, it is going to our food plate. It means soil, a plant is not healthy, vegetarian and non-vegetarian diet is also not healthy. That is the reason something is going wrong. And another situation is there with the soil system globally. It is not a matter the country, it is the globally. But right now we are crying for the sustainability, regenerative, this and that, because everything is linked with the soil system. Biodiversity is there, pollution is also I think, yeah. So pollution is there, malnutrition is there, climate change issue is there, food, because right now, if we think about the food security, food security means availability, accessibility, and affordability. Government have many plan for the food security. It means we are not suffering from the food, but we are suffering from the food quality. So it means nutritional security is very important, or it will become with the soil system. Yeah. So even we are standing at the very poor position for the at the global level for the food quality and hunger index, and even the Apex committee, in front of the Apex committee, government put some data. So 69% deaths of the children due to the malnutrition. It means we are not getting good food quality. It is because of we are contaminating the ecosystem, and due to that contamination of the ecosystem, negative correlation is there. Soil biodiversity and sustainability, whatever is there. So everything is not good with the food chain. So that is the reason we think about it. And it is because of the agrochemical market and environmental pollution. Because environmental pollution we can manage with the agriculture system. And here you can see example. Earlier we quoted the example of the Punjab, because Punjab is the hot spot of the cancer. But right now many other states, or Kerala and other states, and from the northeast also, getting craze to how can we will manage the food system, because food system is totally contaminated. That is the reason, not a matter about the cancer, many type of diseases related to your respiratory system, related to your heart system, and you can see many examples in the society. Different type of diseases are coming, because we can say whatever feed food perfectly, not medicine. If you will feed food like that, a medicine, no need to eat the medicine. That is the reason we must be manage our food system, because globally 42% food is people are not getting healthy food, but in Indian we are 74.1% persons are not getting healthy food. Even another story is also there. We wasting the 74 million, whatever we are producing, 74 million ton of food we are wasting. So my point is here, we are putting continuously contamination of the soil and biodiversity, water, and whatever system is related to the soil, it is contamination is also there. Even 74 million ton of food we are wasting. It means that quantity of food we can fulfill the requirement of any European country. So my point is here we are wasting 74 million ton food. It means we no need to cry for the, it's my personal opinion, for the food security. We must be think about the how can we manage the food system, how can we manage the waste, how can we manage the soil, how can we reduce the fertilizer dose, how can we get the quality food, and other than this, how can we manage the resources. So with that aspect, we can manage all these things. Like that, through the wasting of the food, where many type of wasting of the food. Now we are linking it up with the whatever the relation of the food waste, because just I quoted example 74% food we are wasting. So if we manage that food waste, we can manage the malnutrition, we can go for nutrition security, environmental security, reduce water footprint, because like that I am quoting example from the rice. To produce the 1 kg rice, for 3,000 and 4,000 liter water is also required. And sometimes if we are wasting, so how much, not a matter about the water, how much fertilizer, land, manpower, energy, whatever we are wasting. So we can reduce the all footprint, carbon, nitrogen, water, energy, whatever is there with the same, because from the food system, 37% emission from the globally from the food system. Even in agriculture is the good solution to manage the climate. I will quote the some example from the coming slides. Like that, here I am also mentioning, if you produce the like that nut to it is based on the 100 gram of, because protein is the important component of food. So that's why reason I am taking an example from the food. If you produce the 100 gram of protein, so if you go to up to the beef, 50 kg CO2 emission is also there. So we go for the smart management. So that is the reason. If we are wasting the lot of food, how much emission we can manage? So my point is here, we can perfectly produce whatever the quantity, it's okay, but as well as we have to manage the waste, because lot of waste is there. And to produce, behind the production of the different type of food component, lot of emission is there. Like that, to produce the 1 liter of milk, almost close to 10 kg CO2 emission is also there. Other than this, to produce, just I quoted example, to produce the 1 kg of the rice, 3000 to 400 liter water is required. But from the rice field, 30,000, more than 30,000 kg CO2 emission is there from the transplanted rice. So my point is here, to manage the waste of food, to manage the fertilizer, because more fertilizer input is there, more emission is there. To manage the food, we can say pharmaceutical sector or medical sector. So sometimes we will go to the doctor, give the one medicine to particular disease. Other than this, give the many type of medicine as a food supplement. Right now, new craze is there called to biovitamins. So if we get perfect food, no need to go to that type of supplement. So two big issue right now, climate and food. And right now, you can see the example of the CO2, which concentration of the CO2 is more than 400 ppm. So what is the relation from the, yes, CO2, if more emission is there, like that, some new business has been started in the country. Like that, I am taking example from the Delhi. New business like that, oxygen bar has been started. Go to for 15 minutes and pay about 500 rupees to take the fresh oxygen. And many type of disease, because if you are not getting fresh oxygen, many issue is also there. Other than this, high concentration of the CO2 in the atmosphere and many more greenhouse gases, lot of fluctuations in the environment, different type of, somewhere we are getting drought, somewhere we are getting flood, somewhere we are getting fires in the forest, and some year like that, snow is converting to the melting and getting rains. So sometimes like that, in Kashmir or temperate region, many times we are getting floods because snow is converting to the, it is because of the temperature. So what is the solution? We like that in agriculture. First of all, agriculture is the backbone of the country, and we go to the net zero emission. Because under the United Nations umbrella, we already coded, or we can say, signed on the agreement, we will reduce the greenhouse gas emission. So different of CO2 emission is there. Even CO2 and carbon and organic matter is the important component to the soil system. It is linked. Carbon is totally linked with the all health. Like that, carbon means organic matter. If organic matter is more in the soil, it means soil system will be, or soil biodiversity will be good, because macro or micro, many types have fauna, flora is also, the food is carbon. If microbes are getting good carbon, soil quality will be good, and organic matter is physical, biological, chemical, we can say properties also increase. If soil system is, or soil function and biodiversity is good, plant health will be also good. And if plant will be taken by the vegetarian and non-vegetarian diet, definitely if sometimes we are taking meat, and meat also coming from the plant, it is a food chain. So my point is here, so with the one component, to manage the carbon, everything we can manage. We can manage the human health, we can manage plant health, animal health, and environmental health also, because we can capture the CO2 in terms of carbon sequestration. So that is the important component for the One Health mission, because whatever food, and 95% food we are taking from the top layer of the soil, 15 and 30 centimeter. And if top layer will be healthy, whole ecosystem and planet will be healthy. And even more CO2 emission from the agro ecosystem, we can again capture it to the top layer in form of the carbon sequestration. And carbon is organic matter. Organic matter means like that we are crying for the organic farming. If soil function will be good, need to the very high amount of the fertilizer. So we can, so my point is here, we can manage the environment in terms of the carbon sequestration. we can manage the soil health, plant health, animal health, and environmental health apart from the upper top layer of the soil. For that one, soil carbon management is important because soil carbon is connected with the every ecosystem services. So ecosystem services will be healthy with the carbon management. So we can regenerate the soil with the carbon management. So many type of stress we can manage with the carbon. While in Indian condition, Himani, just you click me if time will be short. I will skip some slides. Because in India, many, if you go to the whole scenario, according to the Center for Science and Environment, New Delhi report saying 85% soil of the India is deficient from the carbon. Because we are tropical country, we are getting oxidation fastly, and after green revolution, many type of intensive tillage we are facing. So we must be restore it with the carbon, and we can regenerate. One my colleague Gahira is also here. So regenerate the soil with the carbon, because if we regenerate the soil, food system will be regenerated, and we can avoid the very much input of the fertilizer. We can totally not depend on the biovitamin and other supplement of food. So sometimes we can say some things is there. Regenerate means something we are giving to the soil. Sometimes we cry for the sustainability. Simple definition is there. Use the whatever resource is there and put to the and leave it for the future generation. But who is the responsible after green revolution? Very many type of contamination is there. Very much contaminated food system right now. So we must be go to the regenerate, and just we are settled down on the organic. Just not a matter about the organic. We think about critically in organic. Whatever in system in the organic, it will give something to the soil. And conventional agriculture is of course related to that. So another example is there. In conventional agriculture, we are in the soil system, but in regenerate in the soil, something will be also we are paying to the soil and we are adding to the soil. And it is a different type of steps of go to the good food and environment system. Here we are here with the chemical farming, but we can manage the sustainability with the NRM, natural resource management, but we regenerate soil for net zero emission, and right now term is go for the net negative emission is also there. It can be managed with the because agriculture is the one of the sector, it can be capture the CO2. Otherwise, if, automobile, whatever the daily life facility and industries, all they are emitting the gases. I am lucky for the science professor. It was under him. He gave the concept 4 per 1,000. It means in the 1,000 gram of the soil, we add the 4 gram of the carbon. So in the coming 20 year, every things will be sorted out. It means you will get healthy food, you will be get healthy environment, all these things. But we needed to implement the plan to purpose, because that concept was the pillar for the Conference of Parties 21 in 1916, I think. So, and upper top layer of the soil is managing the all these things. So my point is here on the top layer of the soil, not a matter to add the carbon. It is a matter that we stabilize the carbon. So how much stability, it is more important than whatever you adding, because whole texture and structure and whatever the physical condition of the soil will be changed by the organic matter. Because in the agriculture, it is big challenge. We are whatever we are putting, it is going to oxidize. So we must be stop the oxidization. As per my personal review and experience, working for the 5 years, we are just adding the maybe in organic farming we are adding, and that is the reason sometimes we have conversion period in the organic farming. But point is here not a matter about the adding. We must be go to the stability up to the depend on the whatever type of practices and input you are giving. So we can take that type of practices. We can say carbon will stay more in the ecosystem. And right now to manage the carbon, another term is there in the society, carbon credit. Of course, by this date, there is no perfect policy with the government of India. I will quote some example in the policy slides. So there is no perfect plan. How can we generate the carbon credit? How can we pay? How can we manage? But many private sectors already started, because under the United Nations umbrella conference of parties, we are committed to, we can reduce the carbon credit. So it will be comes from the we can manage the natural resources, because while in Indian condition, to manage the natural resources, we are very poor to conserve the natural resources. We are standing 176 out of 180. It means we are very poor to manage the resources. That is the reason we are wasting, and we are right now we are need to perfect. Many organizations are doing outstanding for that one, but we must be involved the farmer. Farmer will take the initiative. We can reform the training program to the farmer. So through that one, we can conserve natural resources, and we manage the carbon stability. Through that one, we can generate the carbon credit. Other than this, I'm quoting an example here, the industrial waste management. With the industrial, because industrial waste, especially those waste, are safe for the food system, not contaminated with the heavy metals. If that type of agro waste and other food waste we will convert to usable, so we can definitely reduce the fertilizer consumption. So it will be also increase the soil health condition. So that is the reason we go for the perfect crop production. Because my time is, I think, short, so I will skip some slides. How can we generate the carbon credit? It's a simple techniques and simple game. Pay something to capture the carbon, especially those sectors, those are emitting lot of carbon. They must be go to the space almost net zero. But how can they will go net zero? Because they have industry, they are just emitting. So they must be purchase the carbon credit. So it is a new era in the market. And the like that share market, carbon credit market is there, and carbon credit market is and up. So it can be generated with the 172 type ways to generate. It depend on your what area is there with you generate the carbon credit. So it is different methodology according to the IPCC, and many agencies are developing their own methodology. How can we are also analyzing the carbon credit under the DST project, and we are getting some good, not a matter just putting the carbon and producing the crop within that umbrella. We can say how can we generate the carbon? It is very important. So we are analyzing, and we also explore the whole maybe 4 districts about the what is the situation of soil and agriculture. With that one, we can generate the carbon credit and go for sustainability, and it is one of the key and important sectors. Agroforestry. With the agroforestry, we can manage many things: food security, nutrition security, soil security, environmental security, many security with the agroforestry. So agroforestry is the key player in the ecosystem. So to manage, right now we are agriculture country, so we are not very much focusing on the agroforestry. But right now, under the climatic changing era, everybody is taking interest to go for agroforestry system. That is the reason UP is the first government to implement the policies related to carbon credit from the agroforestry system. And government is also going to plan many big stock sort for that purpose. Another study is there, we analyze from the agroforestry system. So we can, if we involve the trees, definitely we can get the more carbon credit. So some examples from the different agroforestry based models. So we generate the carbon, how much quantity we can capture the CO2 from the atmosphere, and we convert it to the carbon credit. As well as whatever the such system is there, we must be diversify is there. So through that one, we can capture the CO2, and through capturing the CO2, we can generate the carbon credit. So different type of model is there, and you know very well which trees very much we can say a highly biomass accumulating tree and converting to the carbon, because tree have 40% carbon almost. It is depend on the species, and crops have on the basis of the biomass 40%, and trees about up to the 50%. So we can generate it. And in the agronomical approaches, through the agronomical approaches, we go for the reduce the CO2 emission. Through reduce the from the agriculture management, we reduce the CO2 emission, and we convert it to the how much we can reducing from the agriculture practices. Those are listed practices through we can generate the carbon credit. I'm taking some steady examples from the like that Nature Pharma, Indigo, where those companies are at the private perspective giving already to the, I think somebody is disturbing here. Like that Grow Indigo is also giving some benefit about the carbon credit to the farmers. In yes, Haryana and Punjab, Grow Indigo and Vera is also doing. So these are the companies, those are working to generate the carbon credit globally. And question is there, how can we pay for it? So it is a measurement, reporting, verification, and GHG emission. How much we are managing reduction, and we convert different type of software is there, different type of methodologies there, IPCC and many other these things. But here I am putting another one my point. We are paying the lot trillions of rupees subsidy. We are paying the for health purpose lot of amount. Even individual household paying lot of amount to the health. Even medical, LIC, whatever is there. But my point is there, if we'll think the carbon, to manage the carbon to the soil or food system will be also healthy. So we must be think about different taxes we are facing. Like that, if you are government servant, almost 30% tax is there, 18 or depend on the product, some GST is also there. But still in the country, we do not have carbon tax. So my point is there, some amount of the carbon tax will be put to the products and to the companies and whatever is there. If you are buying the carbon, many countries already started. And that amount diverted to manage the soil system. With the carbon, promote the organic matter to the soil. So it will be help to reduce the CO2 concentration in the atmosphere, increase the soil function, plant function, vegetarian and non-vegetarian diet, or as well as not a matter about the food security, it will be matter for the nutrition security. So that is the reason we reform the plan for soil organic carbon management. It is very important. So if restoration of the carbon in the soil, definitely, and we involve all these things from the different. And we must be think about the policy, because still there is no perfect policy. How can we implement it? Financing for, of course, some project is also there, but there the critical thinking is not just we are trying to do the project, we increase the carbon, do the this and that. And recycling at the farm level, it is very important. And motivation to for the carbon, if we will like that divert some amount to the farmers, they will take the interest. We will give the training about the carbon, not just a matter, the organic matter, we can say produce the vermicompost, FIM, this and that type. And awareness is there, because still we are not aware about the what will be happening in the future. So it is very important. If we will go for that right and include our country policy, definitely many type of situation. That is the reason we are getting forced to go to that direction. So Ministry of Environment, Forest and Climate Change is also released about the how can we generate the carbon credit. So many gazette notification is there, but as well as Ministry of the Power, through how can we manage the carbon credit. It is very important. So the country organization are working, and these discussion in the pipeline, how can we will generate it. And the policy, because policy will be different, like that public to public, public to private, everybody will be involved in this one. And we must be think about the electricity, biomass, and whatever coal emission, whatever link and divert the money and link the money to each and another sector and go to the perfect sector for that one. So energy sector is the one of the important sector. So my point is here, we must be make a chain: food, energy, carbon, water, economics. That when we will go to the sustainable food system. So these are the major technology and policies and sectors through that when we can generate the carbon credit. And whatever the potential of the to generate the carbon credit or not, doesn't matter if you are sometimes not able to generate the carbon credit, if we will increase the carbon to the soil system, many more benefit is also there. So that and ICR is also working in the direction with the many type of meetings and agendas. So ultimately, I'm going to conclude. Think about involve the farmer in NRM. Must we focus on one health mission? So from one component we can manage the all health. Need to divert the subsidy to manage the ecosystem service. Improve the farmer living standard through the incentive payment for NRM. Eco-intensification, integration approach, understand the role of carbon in food, nutritional, economic, and environmental security. Soil carbon and credit involved in the national policy. So it is very mandatory and demanding for the time. Need to focus on soil stabilization, not just adding the carbon. This is my team. So I salute to Professor Ratanlal and thanks to my colleagues and as well as Professor Gahira and Banerjee and Dr. Sandeep is also working with me. So these are the strength to work like that work. It is so my honor. I am working with this team, and I acknowledge for enrich me and funding. And your university is one of the important component with me. So if you will visit in the Varanasi, most welcome. If you have any questions, so I will be happy to answer.
Thank you, sir. Thank you for your knowledgeable and insightful presentation. And we have enjoyed it a lot. Our participants also have enjoyed this presentation a lot. And participants has asked that how they can contact you. They are so much impressed with your work that they want to contact you and want your guidance in their professional journey. So if it is possible for you, then you can share your contact details in the chat box so that participants, if they want to contact you, they can
most
contact
welcome because we are working for the scientific community, and thank you so much. For I think I finished my task. Maybe some little more minute is also there, but if anybody want to connect me, you are most welcome on my mail and whatever the process to connect. I will be happy to reply if anybody have question.
Yes, sir. Thank you, sir. And your presentation was indeed very beneficial for all of us. So there is a question that what are the challenges we are currently facing in the policy making in a country like India, or specifically in India? What are the challenges for regulatory challenges and how we can overcome them?
So just you repeat it again, please.
So, like, what are the challenges in a country like India we are facing to develop some regulations for this carbon, for reducing this
Yeah,
carbon
because
footprints?
first one thing, we are a tropical country. That's why reason we are right now we are not involving the soil. But in the coming time, many countries are thinking to involve. But we are involved the soil with the like that. I was in US Dharwad, so they are giving the carbon credit with the biochar. And many private sector is also taking interest in that. So I quoted one slide, many private agencies is there. But country is working in that direction. Whatever gazette notification I showed during my presentation, these from the last year. So I think country is also thinking in that direction, and Ministry of the Power and Ministry of the Forest and Environment and Climate Change and ICR is the big player. So I think shortly something will be come out with the perspective. So just I compiled one article in the advances in agronomy. So honorable ex-DG and present DG is also there, and many good authorities is also there. So whatever the possibility to generate the carbon credit, and not a matter to generate the carbon credit, if you manage the carbon credit, many things will be sorted out. So right now it's under the pipeline, but there is no concrete on the platform from the side of government. Even private agencies, those agencies are interested because like that I am holding one agro industry, and any type of industries I am emitting lot of carbon. But in the coming time, I have need to pay for that. So I'm launching separate branch to generate the carbon credit, and many private agencies are interested with the farmers to pay to the farmers, but the hurdles is there. So many things is not clear. It is some clear guidelines from the IPCC, some recommended from the different type of agencies like that are coded example. Those are already working in even India is also. But still lot of needed to work and reframe the policy. So still this is awaited. Just as per my initial, I shared my thoughts being my works, because I am not a policy maker, I am an academician. So whatever I am thinking, I expressed.
Yes, sir. Sir, we totally understand, but as you are working in this field, so we are just getting, we are just try to getting an idea about all these things, because this is a very hot topic from.
So I already
couple of
shared my idea during
Yes.
presentation, whatever the possibilities.
Yes, sir. Yes, sir. Sir, as we know that today is the era of digitalization, and AI is all around us. So, how is it possible that with this technology and AI and somehow with the help of digital agriculture, we can
Of
reduce
course, yes, of
footprints?
course, because you know it is type of tool. Proximal sensing is there, and maybe remote sensing is there. So different and more, many more is there. So through that, when we can get the perfect information, but it should be farmer friendly, because sometimes farmers are not able to purchase it. So it is a matter about the government. How can they will make it the farmer friendly? It is important. So any technology, those are not perfectly, we can say, applicable at the farmer field. Sometimes research aspect is different and practical aspect is different. But of course, countries also working in that direction, and many good technologies is also in the market to analyze the. But it is depend on what is the aim of is there. So AI is the perfect tool in the coming time. Not a matter about the agriculture in each sector also.
Yes, sir. It's all depends on how we are using it.
Yeah,
So,
yeah.
yes. So yeah, somebody has asked, sir, what are your thoughts on integrating silvopastoralism in carbon credit and sequestration?
I think silvopasture. So it is good, but you know, first of all, we make it baseline. Without baseline, nothing is possible. So when we will do the agreement, but you know, we can compromise the whatever the first priority is. Carbon credit is the second priority. First priority is the production system, productivity, and food system. With that one, we can manage all these things. So definitely we can. So in sometime like that, I'm coded one example about the agroforestry. Agroforestry is the good sector to generate the carbon. So just we identify those trees. I said think globally, act locally. So whatever the perspective of the globe and whatever, because most of the foreign companies are more interested in it. But according to the locality, which species is there, which type of component involved in the particular land or geography, so that when we can, but definitely it will be, we must be go for the baseline as well as whatever the we are involving in the different of component of the agroforestry system.
Okay, sir. Thank you. So one thing I want to ask that not just like professional, not like academician, not like scientist and PhDs. I just want to ask a question from the perspective of Aam Aadmi of India, the common man of our country. Like how a common man of our country, his behavior can influence the carbon footprints of products. Like for general public, this is
Yeah,
a
yeah,
question
of
that
course, because you know, just like that I quoted example, still we are giving the training to produce the vermicompost, to do this and that, like that. So awareness, individual awareness is very important. So right now we do not have plan. Even with that technology, even I am also involved. It is so many scientists are not clear yet how it will be applicable. So common man is the different thing because they are not expert from the carbon. So that duty is by the authorities. So how can we make the plan? It's a matter of the policy because like that I was in US. So one farmer asked me, sir, which technology perfect to sequestrate the carbon during the farmer field visit. So they are very much aware about the carbon sequestration like that, but still we are not aware about even not aware about the carbon. So it is another matter how the other public is also involved. So it will be with the camping, trainings, and some plans will be launched by the government. So it is possible. If like that, I quoted an example to produce the 1 kg rice, 3 and 4,000 liter water is also required. Like that, I'm wearing a shirt for 20,000 liter water is also required. I quoted the example with the 100 gram of protein, how much emission is there. Sometimes we are wasting the food. I quoted the example of 74 million ton food we are wasting it. So if we are not aware about it, definitely that is the reason we are so behind about the target to conserve the resources. So it is because of your mindset, because we are not bothering about it. So it is we can say per capita saving is there, so per capita production is there. So like that, it is we can say joint venture. If everybody is think like that, so definitely something will be good to the country. Many countries have already started in that direction. How much, how much emission we are also less responsible. Just we born on the earth to exploit the resources. But what we are giving to the nature, so we never be bother about it. अगर कभी किसी को कहेंगे तो वो कहेगा ठीक है मुझे नहीं पढ़ाओ, ये इट इज माइंडसेट. तो ये अपनी भी ड्यूटी है. तो इसके लिए हमको परफेक्ट ट्रेनिंग और माइंडसेट चेंज करना पड़ेगा. जैसे अभी इस पे मैंने प्रेजेंटेशन दिया तो आप या जो भी है क्वेश्चन कर रहे हैं, तो जब कोई चीज सुनेंगे तभी उसको, अब इस तरह का अपने पास कोई कंक्रीट प्लान भी नहीं है. वरना एजुकेशन सिस्टम इज आल्सो वन ऑफ द बिग सिस्टम. वी पेनिट्रेट दिस टाइप ऑफ एक्टिविटी, वी रिफॉर्म द, of course we are going to the new education policy, but we are not going to reform the whatever is needed according to the situation. And other countries are also giving the training to the kids about the what is the carbon, how can we manage it, how can we make the resources in school education from the nursery. But still it is not a matter because it is not in my domain. So just you ask me, so that's the reason I'm trying to clarify.
Yes, sir. Very rightly said that we should teach it from school level, and we should make the future generations and every citizen of our country aware about it. Because once we adapt all these things and include all these things in our daily habits, then only we are going to be able for the reduction of these carbon footprints. And in this context, I want to add on that I think we should all accept the concept of minimalism, whatever and how much is required. We should go for that only, and not for the show of things and accumulating lot of things which are ultimately contributing to increasing the carbon footprint. So thank you, sir. It was indeed a very wonderful and knowledgeable session
We are
from
not
your
saving
side.
the resources, we are saving the money for the generation. So if we will not change the mindset, definitely we will suffer it. And many
Definitely,
examples are coming to the with the weather and other direction.
Definitely, sir. And I think most of the participants, they have got lot of knowledge, and I think they may make some changes in their day-to-day life and contribute their bit in this. So thank you, sir. Thank you for your wonderful talk, and thank you for joining us today.
Thank you so much, and thanks to all for giving me an opportunity.
So the participants, as we have, as I have informed you earlier, that Dr. SRK Singh has got some important assignment to attend, so he was not able to address us all at that time. So now sir is back with us, and sir has joined us again. So I would like to invite Professor SRK Singh, Director of ICR Atari, Jabalpur, Madhya Pradesh. So a little bit about sir. So sir has obtained his bachelor degree in agriculture from Institute of Agricultural Sciences, Banaras Hindu University, Varanasi. Sir has completed his MSc and PhD in Dairy Extension at National Dairy Research Institute, Karnal. Sir received merit scholarship during the entire academic period, and during 1999, Sir has topped ARS examination and started his career as scientist of agriculture extension at ICR Vivekanand Parvatiya Krishi Anusandhan Sansthan, Almora. Sir is recipient of 12 awards, including SEE Fellow Award, ISEE Fellow Award, Best Mobilizer Award, Best Extension Scientist Award, Best Extension Professional Award, Young Scientist Awards, Agriculture Today Award, along with Dr. Vasant Rao Naik Award for Outstanding Research and Application in Dryland Farming System in 2021 from ICAR. Sir has numerous publications, including 130 research articles in various national and international reputed journals, 18 books, 53 bulletins, 16 book chapters, etc. And presently, Sir is engaged in research and extension activity with the KVK system of ICR in Central and Eastern India. He has been instrumental in initiating and implementing concepts like KVK KMA, KVK Ring, KVK Technology Park, KVK ATMA convergence model, methodological initiatives for extension research, Nutri-Smart Village, agripreneurship, etc. Sir is also member of various important committees at ICR and state agriculture universities. So the contributions Sir is making to Indian agriculture and the KVK system of ICRs are really commendable, and we are very thankful for Sir that despite his busy schedule, he has taken out his precious time and joined us again for addressing all of us. So, sir, without wasting much time, I would like to invite you to address us all. Over to you, sir.
Thank you, Dr. Himani, for nice and elaborated introduction. And first of all, I thank to organizer, Dr. Mehta and team, to have such a nice platform to share our views and to meet such a new and nice people at this single platform. And I congratulate to the organizer for thinking on such complex and concurrent topic that livelihood and environmental sustainability. It is the one of the challenging area which we are really facing. On today this occasion, Professor P.P. Singh Saab, I have seen the CRP and other things he has shown very elaborately, and really it was a good kind of molecular biology and biomedical research part. Further, Hanuman Thappa Saab, I have listened his words and views. Really, it's encouraging, and definitely our audience might have been benefited from all the kind of words he has spelled out. And then our Professor Girish Chandel Saab and Divakar Saab, Saxena Saab and Tripathi Saab, and all dear audience, the topic you might have gone through, that it's a very elaborative agriculture, animal husbandry, and allied sectors, and then further the livelihood and the environmental sustainability. Three component, clear-cut component is that environment, then it's the sustainability and livelihood. Finally, because mankind always worries about its livelihood. So if anyone, one of us is working on this topic, definitely they are trying to do a very important social service for this nation, in my view. Other may differ from this. And moreover, agriculture is a very vast subject. And nowadays, if we see the future agriculture, tomorrow's agriculture, definitely we have to change the paradigm of thinking first, that just seed, fertilizer, water, this, that only input intensive can't work. We have to think that how methodology based agriculture, how the latest requirement. Now we are switching over to the carbon farming, from carbon credit to carbon farming, rather, that farmers can get the revenue, get the income from even working on the carbon farming, which 10 years earlier we were not able even to think but now we have practiced. Likewise, water footprint, because water is one of the challenging input, and we have to care about water for tomorrow. Otherwise, it's a scarce resources, and agriculture can't survive without water, and human or animals also can't survive without water. So water is another limited inputs for which we have to care. We have to think over the harvesting during rainy season, and then conservation, and then proper utilization. All three things are very in line, and we have to work on those lines which are really suitable for the conservation agriculture. Then a new area of agriculture is coming for the food and nutrition, because if we observe that our country are now self-sufficient in food production, but what about the nutrition? Nutritional gap is still existing, and it's the remarkable gap. So for that also we have initiated, ICR has initiated nutrition sensitive agriculture resources and innovations, NARI. And under that we are promoting the NutriSmart village. In that we are advocating that jo khaye ugaye, jo ugaye wo khaye at household level, because in village all have no purchasing capacity to daily fresh vegetables, fruits and other items, but at least if they grow for themselves, then they can get fresh and the cheapest, as they are growing from themselves or involving the family labors. Third component which coming is the crop residue management. One of the burning area, because farmers are burning their straw. It's really bothering, and it's costing heavily on the environment. I am using term heavily, though it's farmers are not considering it much important, but for us it's important, and for environmental sustainability it's a very important aspect. It's not only disturbing our local climatic condition or weather condition, but also soil microbes are being disturbed. Soil microbes are being destroyed by the burning the straw. So we have to think on these lines if we really concerned with the environmental sustainability. Further, we have to think over the group approach. We must think on the linking the people, aggregating the people, so that we can help the farmers in purchasing the inputs as well as the selling the outputs. Because one of the major issue farmers facing in the ensuing season, that how cheaper they can get the quality inputs. And after production, they are searching that where they can sell their products at the optimum or even higher cost. So if we will work on the group approach, definitely it will lead to the sustainability other than the environment, and also it will help in the searching the livelihood. So new dimensions of the agriculture are waiting to be tapped, to be emerged, to be used, to be promoted among the farming community, so that we can address the tomorrow's issue with latest solutions. Thank you very much for giving me opportunity. Thanks to the organizer, thanks to the Mehtaji and his team. Thanks a lot.
Thank you, sir. Thank you for addressing us and sharing your views about this topic. So thank you so much, sir. Thank you again. Sir, you are on mute.
Thank you, sir.
Yes.
Okay.
Okay, welcome.
Thank you, sir. So, dear participants, now moving forward, we are going to start our technical session. So, as you all know that we have two technical sessions today, technical session 1 and technical session 2. So, the participants whose names in the technical, so the participants whose name in the schedules were mentioned in the technical session 1, I'm requesting to all of you to stay with this link only, and the participants whose name are under the technical session 2 for today's program, they are requested to join the second link which has been shared with all of you. So participants whose presentations are in the technical session 2, kindly join the technical session 2, and those whose presentations are under the technical session 1, kindly stay in this technical session only. We are about to start the technical session, and we are just waiting for a while so that the participants of technical session 2 can join their respective session. So did participants, our chairman and co-chairman and reporter for technical session 1 has joined this session, and they are about to begin with the session. So once again, I'm requesting all of you, if somebody from technical session 2 is joined in this session, they are requested to join the other session, and the link is shared in their WhatsApp group. And I'm also trying to share the link of the technical session 2 here so that you can directly join that link. So dear participants, the link for joining the technical session 2 and meeting ID and passcode is also shared in the chat box of this technical session. So if anybody is here in this technical session, I'm requesting all of them to join the technical session 2. And for the participants of technical session 1, we are about to start. Please wait for a while. So participants, I would like to welcome the chairman of this technical session 1, Dr. Nisha Rana. So ma'am is in charge IPR cell, startup and incubation cell, life science station, Swami Vivekanand Subharti University, Meerut, and madam is the chairperson for this technical session, and ma'am is joined with the co-chairman of this technical session, Dr. Asha. She is also faculty of Basic and Applied Science, Madhav University, Rajasthan. And reporter for this session is Dr. Nagendra Kumar Lohia, faculty of Basic and Applied Science, Madhav University, Rajasthan. So I welcome you all, and I would like to hand over this podium for you for this technical session 1. And in case you need my help, I'm there for any assistance. So you can let me know. And so shall we proceed, madam?
Yeah, sure. Thank you, Dr. Himani. Yeah.
Thank you, ma'am. Yeah. So over to you.
Thank you. I would like to welcome my co-chair, Dr. Asha, and reporter, Dr. Nagendra Kumar Lohia ji. So for this session, we are having a total of 40 presentations. So I'm inviting the first speaker, Miss Priyambi Palai. Priyambi, are you available? Miss Priyambi Palai. Priyambi Palai, have you joined the session?
Yes, ma'am. She has joined the session, but I think you have the rights of host, so you have to make the participants co-host so they can unmute and present their session.
Priyambi, now you can share your presentation. I have unmuted you. You can share your presentation.
Yes, ma'am. Yes, but I think for presentation, you have to make her the co-host.
Okay. Priyambi, now you are the co-host of the session.
Yeah.
Done.
Now Priyambuda can now share her screen.
Yes, ma'am. The screen is visible.
Priyambuda, you are not audible and your screen is also not visible. Yes, your screen is visible. You can put it on full screen mode. Yeah, it's perfectly fine. Now you can start your presentation. You have total of 5 minutes.
Yes, ma'am. Good afternoon to everyone.
You need to speak a little bit louder.
Yes, ma'am. Good afternoon, everyone, distinguished guests, and my fellow experts. I am Priyambada Pallai, currently pursuing my master's degree in agricultural meteorology. As an agricultural meteorology student, today I will be discussing the impact of urbanization on the heat island and exploring the key drivers and its consequences. So let's examine the complex relationships between the urban planning, the impact in the climate, and the human and its well-being, health well-being. So this is my flow presentation. Come to urban heat island. So the thing is very simple. When the urban experience more warmer than its natural temperature, it is known as the urban heat island. And nowadays the main cause of the urban heat island is the industrialization, more infrastructure, and the population. And another three reasons, that is the heat absorption, heat retention, and the lack of vegetation. So basically in concrete area, that is low albedo. So for this region, low albedo, it absorbs more solar radiation. So urban heat more, and lack of vegetation, and heat retention, that more building, so they are close to each other, so reduce the air circulation. These are the classification of urban heat island. One is the surface and another is the atmosphere. The surface, which is the day temperature 18 to 70 degree Fahrenheit and the night temperature 9 to 18 degree, and in the atmosphere, which is less variation than the surface UHA, and we can measure the surface UHA by the thermal image and the atmosphere by the isotherm gas. These are the causes which increases the urban heat island. Basically, the CO2 emission, heat release in the atmosphere, less cooling efficiency, less convection, heat trapped in the atmosphere. These are some of the causes. So, effect of the UHA. Simply, I am saying the effect of UHA. The people demand more electricity nowadays. So that's why the more power plant needed. For more power plant, the extra energy also needed. So for this reason, more greenhouse gases is emitted. So ultimately, it causes the global warming and the climate change, which will affect the environment. And it is the how the which I said before, how the urban heat island which affect the greenhouse gas emissions and the air pollutions. And the main sector of the contribution of the greenhouse gas emission highest in the case of 6 to 18% in our agriculture, and some of the gases, that is the carbon monoxide, carbon dioxide, sulfur dioxide, and nitrogen oxide. How the urban heat island it is danger to the aquatic system. If the temperature increases in the rooftops and the pavements, temperature also increases. When the runoff or drainage water flow, it will flow directly to the river, the pond, lake or streams, which causes the thermal pollution and ultimately affect the reproductive and metabolism system of the aquatic systems, aquatic animals. These are some of the secondary impact of the weather and climate. There are two pictures in this slide. The first picture of the image, which is the flooding area, and with some of the people wading through the water, which representation the impact of the climate change. And another picture, which is the drought-stricken area with a person sitting in a dry field. So there are two pictures in this weather changes, these two pictures will affect the urban heat island. And when the urban heat island is more intense or upward wind, then that moment the thunderstorm or precipitation activity occurs. If unusual heat occurs, which is also a cause of the thunderstorm and precipitations. And low pressure area, it draw the cool air and including the cloud and rain form occurs. These are some of the recent news in which the Mumbai experienced the urban heat island phenomenon. So I will say the current situations of the Mumbai, which will experience the highest urban heat island. So now the question is, why Mumbai is so hot? Because Mumbai is present in a coastal area. The city's climate is shaped by its coastal locations and high humidity. But still it is hot. But why it is hot? The main problem is the Mumbai populations. Because as the Mumbai populations grows, the heat occurs and making this weather more worse conditions. So recently, Mumbai's temperature and humidity, which have soared, and in recent news which I read from the Twitter on April 16th, the city hit a highest record of the 49.7 degrees Celsius. This high heat and humidity, which will impact the weather of the Mumbai climate. And for these reasons,
Priyamta, you have to conclude it a little bit faster.
For these reasons, the people use more electricity, that is 4,041 megawatt electricity they use. So it is a problem. And these are some of the government who use for mitigation of the urban heat island. They use the cool roof payments. In Telangana, it is highly effective. And green buildings and also used. I took two two case studies. One is from the geospatial world format. Directly I come to the result part. So according to the LULC classes, in summer seasons, the highest differences in LST, that is the LST means the land surface temperature, the highest in the dense vegetation, and in winters and the lowest in the water bodies. And the average differences in land surface temperature, highest in winter seasons to the built areas, and the lowest in the water bodies or dense vegetation. These are the another case studies.
Priyamvada, please conclude your presentation.
Yes, ma'am.
Go to the concluding remark.
This is the green vegetation. So now let's conclude the presentation. So to sum it all up, the urban heat island are basically the cities turning into the giant toasters. But on all seriousness, urban heat island is not a laughing matter. They affect our health, energy use, and the climate. So, but the good news is we can cool the things down by greening the roofs, more trees, and reflective building materials, which lower the temperature. And another one thing I thought during my preparing the slide, sometimes the best way to beat the heat is to go to the old school, more shade and more plants, so that fewer heat trapping the materials. Thank you. These are the references.
Okay. Priyamvada, this is your review work. Priyamvada?
Ma'am, actually, currently I'm doing my master's degree. It's my work of my seniors.
Okay, you are a PG student.
Yes, ma'am. Yes,
Okay. And what is your contribution in this work? As you have reviewed someone, then what is your contribution in this work as a reviewer?
ma'am. Actually, in Bhubaneswar, in our OET also, we will track the temperature and the weather conditions and how it will affect witnessing.
Okay. Then the next participant is Mr. Sudarshan Panchal. Sudarshan Panchal. I'm searching with your name, but I'm unable to find you. Please.
Yeah, actually, Sudarshan Panchal has joined earlier, but I think he has left, so we can move with the next participant, Mr. Miss Sharon. I'm so sorry, Miss Sharon, and she has raised hand. You can identify her from there.
So, Miss Sharon Monica R, you can present your work. Okay, I'm adding you as a co-host. Sharon, now you are the co-host, so you can start sharing of your presentation. Sharon, please unmute yourself.
Yes, ma'am. Yes, ma'am.
Okay. Please try to sum up within 5 minutes.
Yes, ma'am. Is my presentation visible, ma'am?
Yes.
Good evening to the honorable members and my fellow participants. I'm Sharon Monica, and I'm a postgraduate student from Tiruchirappalli of Tamil Nadu, and I'm here to present an oral presentation on the topic ecological plasticity and metabolic diversity in Iconoclaspis mud storms. So before I could move on into my presentation, I just want to start of how did I come up with this project. As we walked down to the aisles of Tiruchirappalli, me and my friend saw that the streams and canals of Tiruchirappalli was infested by this iconia because we know it's an invasive weed. So my friend asked, as a botanist, what is the step that you're going to take in? So because of that, I've taken the initiative to come up and summarize of how can we eradicate or how can we bring up a control measure in Iconoclaspis by studying the ecotype study. So this is my abstract where I've given where are the regions. I've taken 3 regions for my consideration, and I found out the morphological differences, anatomic differences, then the phytochemical screening of what I've done, then the spectral analysis, the elemental analysis, and the water assessment that I've done in this abstract. So India, we know it's facing a lot of crisis in weed control. So we know that weeds are unwanted plants that can reduce the crop yield and the quality. And to see like in India, there is about 31% that has been exploited by these weeds. And the common weeds that we see are Parthenium, Lantana, and Water. Though there are a lot of steps taken, there are still more problems that are yet to be faced. So, as we study about weeds, we must know that weeds vary according to their areas, according to their variation or the physical attributes. So, one among the variation is seen as ecotype, and we know that these plants adapt themselves to the local environment. So, if we talk about Arabidopsis, it also have invaded itself. It also have adapted to the local environment, making it more prevalent in such areas. So here, as I've taken Iconia, this Iconia is seen to be ecotypic in the region of Tiruchirappalli in Tamil Nadu. So as I studied about ecotype, I have to refer to elementum analysis. So the elementum analysis is the elemental composition that includes nitrogen, phosphorus, potassium, carbon, oxygen, etc. Of a plant or anything else. So the instruments that we can utilize is this ICP-OES, which stands for inductively coupled plasma optical emission spectrometry that I've been utilized. So in my aquatic plant, we seen that Salvia and Iconia crassipes have been invaded West Bengal and Assam. About 115 acres of West Bengal has been infested only by Iconia species. And that is a threat to a human being and the agricultural lands. So it's a gist about Iconic crassipes, which is native to Amazon, and it forms a dense mat that blocks the sunlight, and it retards all the other nutrients that is required by the zooplanktons and any other microorganisms or living organism that is inside the water. So this Iconia reproduces both sexually and asexually, that makes it difficult to control. So the impact that it brings about is ecological and socio-economical. In ecological, it reduces the water clarity, oxygen, then it reduces the phytoplankton, disturbing the aquatic chains. Then socio-economic is it disturbs the fishing aspects of fishermen, then the transportation across water bodies and the irrigation that has been maintained by these agriculture farmers. So thereby it brings about higher maintenance costs for many farmers in India. So there are many management structure challenges and solutions that are brought about, but the best thing is to do is to analyze how it can be done as an eco-friendly way, as an integrated weed management by combining all the mechanical, chemical, and biological aspects. So my methodology goes about like first we identify the plant, whether it's Iconic crassipes according to the flora, uh, and we have analyzed to Rapinath herbarium in Tiruchirappalli of St. Joseph's College. So the methodology that we have followed is we collected Iconia from 3 different regions within
Sharon, focus
Tiruchirappalli.
on your results
Yes, ma'am. Yes, ma'am.
and try to
So
conclude this time.
yes, ma'am. Yes, ma'am. So my results were like seeing in 3 regions, the morphological showed variations in site 1, site 2, and site 3. So in site 1, site 2, and site 3, it showed variations in its leaf width, petiole, and root length. Then anatomical variations, it showed in leaf, petiole, and root, where we could find S1 having more vascular bundles, here S1 having more vascular bundles, and in petiole of S3, that is, we found more sclerites. So we did the preliminary analysis where we found protein, steroids, tannins, saponins present, and UV visual spectrum of 200, 300 nanometer in all 3 sites. FDI findings were like hydroxyl alkenes, alkenes, and carbonyl was present. So this is the chromatogram of UV spectrum of S1, S2, S3, and FDI for S1, S2, and S3. Then GCMS, we found that S1 had the more 18 components, which includes cycloheptasiloxane, which had antimicrobial property, and S3 had 2 important components, that is 12-hydroxysteramide and arenol, which had anti-melogenic property. So this is the elemental analysis. We found out that phosphorus is more in S1, and in nitrogen, S2 had the nitrogen level in more. then water characterization, we found that S3 was more contaminated, whereas S1 was more organic polluted, and thereby the microbial activity was more. So in my conclusion to come up with, there are a lot of things that we found, but predominantly that we saw was using, instead of total eradicates, a sustainable solution to its investigation lies in integrating ecological control with bioutilization approaches like phytoremediation, composting, and extraction of various valuable compounds. Like as we mentioned in GCMS, cyclohexanol and benzamide were present in all three sites, which has antifungal, antimicrobial, and antiperspirant activity. And Iconoclaspis also thrives in high nitrogen phosphorus condition, suggesting that controlling nutrient runoff, especially from agriculture, can limit its growth. So this is our findings in our project work of Iconia in its ecotypes.
What is the significance of your study?
Ma'am, significance like finding the ecotypes.
Perspective.
Pardon, ma'am?
What is the significance of your study?
Importance that of my studies, if we understand the ecotype and variation among the sites or wherever it grows, Iconia grows, depending upon it, we can remove the Iconia, ma'am. If it's having long root, we can eradicate through mechanical support. If it is short, if it has a short petiole, we can use some biofertilizers that can prevent its growth. So depending upon its variation in its ecotype, differences in location, we can identify what bioremediation or what bioutilization that we can promote it with, ma'am.
Okay. Now this is open for question answer session. If anyone want to ask anything, then you can ask.
Yes, participants, if you have any question in between, you can drop your questions in the chat box so that we can ask your questions from the presenter.
So if there is no question, then we can move forward. Then I have received one more request for participant number 22. Nishtha.
Yeah, message I have got from the name of Akanksha Singh. I think they have joined from other name.
Okay. So
yes.
I have to make her co-host.
Yeah, participants, I have a request from you. Please edit your name as your name is mentioned in the schedule so that we can identify you. Otherwise, it will be difficult for us to identify you, and it will waste our time. So I request you to rename.
Akanksha Singh. Now you can present your work. Akanksha Singh.
Hello. Yes, ma'am.
Now you can present your work.
Yeah, sure, ma'am. So good evening, everyone. I am Akansha Singh, a research scholar from University Department of Botany, Tilkamanjhi Bhagalpur University, Bhagalpur. And the topic that I'll be presenting is molecular detection of begomovirus Solanum deliens virus in tomato crops. So coming to this is the content that I'll be sharing. And coming to begomoviruses, these come under the category of Gemini viridae, which are non-enveloped plant viruses with small single circular DNA strands, genome of 2.5 to 3 kilobases. They can either be monopartite or bipartite arrangement. And the most important family among this is begomoviruses. So this is the species distribution of Gemini virus, in which 85% is covered by begomovirus only, and the rest, such as Bicoto, Topoco, Grobalo, these are covered by the rest of the viruses. So coming to leaf curl viruses, tomato is considered to be one of the favorite host for viruses, which has over 50 species naturally infecting them, and the viruses which is clustered under this generic name, which is tomato leaf curl viruses, these cause the most devastating tomato diseases. One of them, which is tomato leaf curl New Delhi virus, is one of the specific area that I'll be discussing. So as you can see in the picture, some of the symptoms, which is upward curling, mosaic, defolious deformation, mosaic symptoms, crinkling, puckering, these are the kind of symptoms that we see in the fields. So coming to material and method, the first is sample collection and DNA isolation. So the leaf sample of from different places of location of Bhagalpur were collected, and their genomic DNA was extracted using genomic DNA isolation kit. Along with that, we also took the samples of asymptomatic leaves, and we extracted their DNA also. Next, we went for PCR assays of the sample. First, we confirmed that if there are any begomovirus is present or not. So we confirmed that with a begomovirus universal primer pair. Once we confirmed that, we went on to choose check between different plant viruses such as begomovirus solanum jeduparense, solanum karnatakense, Delhiense, palampurense, gujaratense, and then Bangladeshense. Once we confirmed that, we went to their primer IDs. We ran a PCR, which was performed in Sure Cycler with a single step of preheating for 3 minutes and denaturation annealing. And annealing temperature was set as per the primers we used, followed by extension for 1 minute and then for a final extension. Once we did the PCR, we went for gel electrophoresis, where 1% agarose gel prepared in tea buffer was used to visualized, and then they were visualized in gel documentation system. Once it was done, we went for cloning and sequencing. Also, out of all the suspected samples, we took some isolate and we subjected them to AV1 gene amplification. We cloned them into PZ cloning vector using CloneJet cloning kit, and then they were sequenced. Once they were sequenced, multiple sequence alignment was established, and then these were studies with creating a maximum identity with viral DNA sequences that were isolated from the samples for pairwise person nucleotide identity. We also confirmed the distribution and incidence of the viruses. So a roving survey was conducted, which was to assess the prevalence of virus. So different locations that we took, we set there 3 plots at each location and an average of 150 plant population. We choose curly stunted growth, and we tend to counted all the plants. So the disease incidence was counted with this formula, which is number of infected plant by observed plants. And then we also went for to study the dynamics between the whitefly mediated transmission in tomato crops. For this, we went to for the several we studied this in several kind of varieties of tomato, such as Punjab Chohara, BRDT, Kashi Vishes, and determinant cherry tomato. We set up an experimental design at the field, and each plot contained 30 plants for the whole chromic periods.
Akanksha, please focus on your result and try to conclude
Okay,
within
ma'am.
5 minutes.
So a total of 85 symptomatic leaves were collected. Out of them, 60 were positive for the begomovirus infection. Out of those 60, 39 were found positive with AV1 gene primer, as I mentioned earlier. And the rest of the tomato leaf curl virus were not found positive. Out of all of these, we set up a PCR assay, and this is the result, where a total of 60 is found associated with whitefly transmitted Gemini virus, and out of which 39 were found positive with tomato leaf curl New Delhi virus. And this is the percent nucleotide identity of AV1 gene between the isolates that we did and other reported gene at the NCBI database. And you can see there 95% identification is found positive. And then the result from distribution and incidence, a high incidence was found at one of the regions, which is the Sabor region. And we also tried to find relationship between whitefly population and tomato leaf curl incidence, which we found
Akanksha, please
positive.
conclude. Your time is over.
Okay. Tomato leaf curl disease, as it is the one of the most important viral diseases of tomato. So we have tried to study we have tried here studying the epidemiology of the diseases and how can we cure all of that. And also we choose to develop a host reservoir program with an integrated control approach, so that it will be beneficial for any kind of agriculture land which practice this kind of promote this kind of common crops. So these are the references. Thank you.
What was your sample size?
Sorry, ma'am?
What was your sample size for this study?
Ma'am, we don't go by sample size. We just collect young leaves which show the symptoms, and then we take around 100 mg of leaf tissue to crush
How
to
many
pick the
plants you
DNA.
have collected? Plants or leaves you have collected for this study?
Ma'am, leaves.
Leaves, for leaves only. How many leaves of plants?
For
For
DNA, so
10, 12, 30. You
okay,
don't have any fixed number of the
yes, ma'am. We tend to collect as, for example, we are taking a 25 to 30 leaf samples for once, and
single plant from different plants.
from
Or
single plant only, from single plant, 15 to 20 leaves, and then if we are extracting DNA, we will measure 100 to 120 mg of leaf tissue for DNA
I'm not
isolation.
asking about your methodology. I'm just asking about your sample size for this particular type of study and for the variety of plants. And number two is the your kit. You are saying about the DNA isolation kit. You have using kit method. Then you are using the kit of which company?
Thermoscientific.
Okay, that means comparatively good. So you don't know the exact sample size of your
Ma'am,
study and
ma'am,
you have conducted the
ma'am,
research experiment.
Ma'am, I will just explain. We don't have a perfect sample size here for viruses study. We just we choose to take a younger leaf. We collect around 20 to 30 leaf, and then we have a fixed sample size that we take for DNA isolation. We don't have a sample.
That was random sampling. Okay.
Okay, yes, ma'am. That is sampling is done.
Random sampling for your study, and then your sample size is fixed for DNA isolation.
Yes, that is 100 to 120 mg.
Okay. Now the next.
Okay, thank you, ma'am.
Next is Khushbu Tyagi. K. Uday Kumar is present or not? K. Uday Kumar, number 5. Miss Pushpa.
Participants can raise their hand to mark their attendance so that madam can identify you.
Miss Pushpa. So the next one is Miss Khushbu Tyagi. Khushbu, you are already added as a co-host. Now you can share your presentation. Khushbu, you can share your presentation. I have already added you as a co-host. Khushbu, sequence number 7. Khushbu Tyagi. So next is Abhishek. Abhishek. Okay. Pushpa is also available. Khushbu, you can start your presentation. We will be adding Miss Khushbu Tyagi after you. Khushbu, please unmute yourself. I have already.
Khushbu is not audible.
Ma'am, I have already added her as a co-host.
Yeah, you have, madam, you have done, but Khushbu has not unmuted herself, and her camera is also
So
off.
Pushpa is also available. Now we can do one more thing. We can add Pushpa.
Participants, once we make you the co-host and your turns is there, you have to unmute yourself and you have to present your screen.
Pushpa, I'm unable to find you. Please change your name.
Please change your name as per the name mentioned in schedule.
I'm calling the next presentee, that is Shweta Dhiman. Shweta?
Yes, ma'am, I am present.
Okay, please start presenting your work.
Okay, ma'am. Good evening, everyone. Myself, Shweta Niman. I am from College of Forestry, Department of Tree Improvement Forestry. Today, my presentation topic is effect of seed source on fruit seeds characteristics and seed germination of Prunus cercis didon. First objective is to study the effect of seed source variation on seeds and fruits of prunus certices from different seed source. Second is study the effect of pre-treatments on seed germination of prunocytes from different seed source. Introduction. Prunus certices Didon is a sacred plant in a Hindu mythology which belong to the Rosaceae family. Prunocytes is one of 31 multipurpose tree species of Himalayan regions. Distribution from between 1200 to 2400 on slope land reaches heights of 10 to 11 meters. Prunus certices is traditional used for edible fruit, seeds, gums, as well as different medicinal application. The leaf of Prunus certices is used to make green dye fruits and used to make a dark gray to green dye and tanning to obtain from the bark. Prunus certices trees produce a large number of seeds with hard seed coat, and the regeneration of this species is a very poor in its natural habitat. Material and methodology. Collection of a planting material on the basis of the survey. 6 sites which population of prunus certices were identified. The study 6 sites of Tiri Gadwal. The first table, geographic information of 6 selected sites in this table. Siliara, Devalgaon, Kuteti, Ranichori, Sadargaon, Charangikhal. And a block, Ghansali, Uttakashi, Chamba, or Pratapnagar, Uttakashi. Second table is a treatment detail. First is a seed with seed coat plus cold water for 24 hours. Second is seed with without seed coat, cold water treatment. In the other treatment, in a 15 treatment in this table. Some picture during my practical work. Morphological study. First is a study on a fruit morphology, fruit length, fruit width, fruit length, width ratio, fruit thickness, fruit weight, fruit moisture. As like a study on a seed morphology, seed length, seed width, seed thickness, seed length and ratio, seed weight, moisture content, pill thickness. Study on seed germination percentage. Second is mean germination time. Third is germination index.
Hello.
Study. Hello.
Participants, please mute yourself during the presentation.
Study of seedling growth, plumel length, radical length, and radical and plumel length ratio, seedling length, and seed vigor length. Statistical analysis of morphological data, analyze RBD form, and lab is a complete CRBD form. Experimental result, morphological study and seed study. First is a morphological studies, and the best result of fruit length in a Ciliara and the minimum in a Charangikhal. A fruit width highest in a Devalgaon and minimum in a Sadargaon. Fruit thickness highest in a Devalgaon and minimum in a Charangikhal. Fruit length in a highest in a Ciliara and minimum in a Devalgaon. Fruit weight highest in a Ciliara and minimum in a Devalgaon. Moisture content highest in a Devalgaon and highest in a Sadargaon and minimum in a Devalgaon. Variation of seed characteristic. Highest seed length in a Ciliara and minimum in a Raichur. Highest seed width in a Ciliara and minimum in a Kuteti. Seed thickness highest in a Sadargaon and minimum in a Kuteti. Moisture content highest in a Sadargaon, minimum in a Devalgaon. And seed length width ratio highest in a Ciliara and minimum in a Ranichuri. Peel thickness highest in a Devalgaon, minimum in a Ranichuri. Seed width highest in a Devalgaon and minimum in a Ranichuri. Some picture during my lab experimental. Variation on a seed germination percentage, different pre-treatment in the seed source. The highest seed germination in a T14 treatment, Ciliara 92, and a minimum in a 55. And the second highest seed germination in a T4, Develgaon 82%. Conclusion, the overall finding of present study indicate that the variation in a fruit and seed size significant influenced by the seed source. The superior fruit and seeds with respect their morphological were recorded near Ciliara, Kuteti, Sadargaon seed sources, which was significantly affected seed germination and seedling growth. The maximum seed germination seedling growth were obtaining in the Ciliara, Sadargaon, Kuteti seed sources. On the other hand, pre-sowing stratification with the treatment GA3 and hot water treatment for 24 hours were significantly enhanced the seed germination and seedling growth, which was more effective as compared to other treatment in case of prunus herbicide. Recommended in generally seed stratification with a treated GA3 is not economically suitable for the poor nursery grower and farmers because lack of resources. The hot water treatment is any easy and cheapest method for mass multiplication of prunus herbicide in a forest nursery and plantation. Thank you.
So now this presentation is open for questions. So any query from the co-chair and rapporteur or any participant? So I have one query. Shweta, what is your future perspective of this work?
Now
What is your
I am a PhD scholar and
no, I'm just asking about your work only.
I continue my PhD work in this topic.
Okay.
In a large scale on a Uttarakhand state.
In Uttarakhand state. So, what are the significance of this study? Shweta?
Yes,
I'm
ma'am.
asking about the significance of your study.
A seed and fruit characteristics and the seed germination and a correlation. That three factors for us.
So the next is Professor Atul Bangari. Sir, you can start your presentation. Okay, Shweta. Professor Atul Bangari. Sir, you are the co-host. You can start your presentation.
Yes, madam. Thank you so much. Thank you for this opportunity. Let me pull up my presentation. Yes. Yes, madam. So good afternoon. Good evening to everyone. I'm Professor Atul Bangari from MIT Vishwavidyalaya University, Solapur, and I've been joined by my student Suresh Gaikwad, who is BSc in agriculture and MBA in marketing. Today's topic that we are presenting is data for the Annadata, which is mainly to see how important data is and how data-driven decision-making can be applied. And that's where we have done extensive market research to come out with how agronomic intelligence can help farmers for sustainable farming. So that's the topic, and adhering to the template provided. We'll start with the abstract. The abstract was submitted. Mainly, what we have done is that we have reached out to farmers across various different locations in the state of Maharashtra through the KVKs, the Krishi Vikas Kendras. First, to identify the challenges that they face, and then to analyze it through correlation and heat mapping techniques, wherein we have applied the statistical correlation analysis to bring about the positive correlation, strong positive correlation, and also some weak or negative correlations. And that is our presentation today. And why? What is the need for doing such an intervention? Is mainly that globally, as part of the Sustainable Development Goals, it's all about to create food security and bringing in sustainability for feeding the populace of the world. So it's a global imperative. It is the need of the hour, and there's a lot of focus that is coming in from not just the farming community, but also the tech community to see how technology can help. And our intervention is through seeing how data-driven artificial intelligence solutions can help in transforming the present day agriculture practices and also address some of the critical challenges that are faced locally. And the most important thing to note here is that agriculture and agriculture practices are localized. And that is where we are talking about agronomic intelligence. Again, it's AI, but it is not artificial intelligence. For farmers, for the agriculture sector, it is the agronomic intelligence, which basically defines the way data can be used about the agriculture. When we say data, it's the data that governs every aspect of agriculture, which includes the weather conditions, the soil conditions, the crop availability, the seed condition, the fertilizer to be used, the seasonal effects, the environmental effects, the weather conditions, the seasonality, so on and so forth, right? So it's quite an enormous task when it comes to handling these many parameters and this much of data, which is where artificial intelligence can be applied. And we all know that data-driven decision-making through artificial intelligence, through the use of artificial intelligence, can basically decide, or rather provide the inputs for the farmers to decide in the right way so that it can have the right impact. So our way to do this was mainly to collect the data from various agricultural districts, wherein we have identified the challenges that the farmers face at their locale, is the location, and also the digital readiness. It's a marriage of these two aspects that we need to ensure. And that's how we collected the data across close to around 8 locations, catering, or rather, we are still collecting data. But for the time being, I think we have close to around 1,600 odd responses. And on that, we have done simple statistical correlation analysis and heat mapping techniques to establish the relationships and the correlations. So what are the results? 2 things. One is the systemic constraints in terms of the rising input costs. So we identified a strong correlation between pesticide, fertilizers, and herbicide costs, which mainly is indicating a systemic inefficiency in the agri input availability for the cost of fertilizers and pesticides. Aside, it's also the access to the knowledge in terms of the government schemes, the government offers, the minimum support pricing, so on and so forth. So while these are some bottlenecks, this is the correlation. This is what we have seen. It's the availability of machinery. It is the rising cost of pesticides and fertilizers, herbicides, across different farming techniques, lack of manpower, climate change, uncertainty of availability of water, and of course, risks of not getting the right advice on farming, uncertain market prices. So if you see, there are some strong correlations and there are some weak correlations. And this is what our finding was or is in terms of addressing some climate concerns, wherein there is weak or negative correlation to the climate change, uncertainty of water, and the lack of awareness or the recognition of climate risks among the farmers. And this is where I think data can help overcome all of these challenges. So to conclude, our finding is that while the smartphone adoption rates are high, the willingness to bring the right localized AI solution is something that the farmers look forward to. And because of the availability of just the smartphone, it becomes a decision support system for the farmers enabled by AI. And needs and preferences is mainly in terms of multilingual interfaces. While we may talk in English here, the local language preference is paramount. So in India, as we say, das goes badle pani, bis goes badle bani. We need to definitely bring about the right language assistance or dual language platforms, wherein we integrate it, which can help in overcoming these multiple challenges of the uncertainty in the input cost, uncertainty in the output cost, and unavailability of the information, market information, government schemes information, and so on and so forth. So the recommendation that we figured out, or the conclusion is, a hyper-local decision support system is needed, and that is what we have developed. We are developing it, and we're going to have pilot with these 1,600 odd farmers very soon, which includes the data integration across all the parameters, the factors, and the components that would help farmers decide on farmers to decide on the best crop, the best fertilizer to be used, the best farming method to be used, the best crop to be sown in a particular region at a particular point in time, in a particular season, or at a particular location. So that is where our work or our research, we expect it to play a role in enhancing the availability of the data and information so that farmers will work towards sustainable farming practices. So that's what it is from our side. Thank you so much for the time. Any questions? I'll be more than happy to take.
Thank you so much, sir. Now the talk is open for question answer session. Sir, your work is basically based on modeling system or
I couldn't hear you, madam. What is that?
Sir, I'm asking that your work is based on modeling system.
Yes, madam. So let me show you the correlation data that you just saw is what we built through our Python program, wherein we have applied machine learning algorithms for predicting and also for correlating, which is what will help the farmers to decide on the inputs that they would provide.
Okay, sir. Any other question from audience or co-chair person? Any question from the co-chair? Okay, thank you so much, sir, for joining us.
Thank you, Dr. Nisarana. Thank you so much.
Next is Ms. Khushbu Tyagi. Khushbu, now you can start your presentation. Participant, please raise your hand so that I can make them co-host. So after Khushbu, I'll mention the name in the chat box. If you are available, you can also mention your name in chat box and announce as per the sequence. Khushbu, now you can start your presentation and please try to complete your Presentation within 5 minutes. Please be bound for time. Vishwavidyalaya, you are not audible to us.
Good afternoon, ma'am.
Afternoon.
Ma'am, am I audible?
Yes, you are audible, but I think you are using two systems for your presentation. That's why it's sounding echo.
Okay, ma'am.
Now you can start your presentation.
Good afternoon to all the respected dignitaries, organizers, esteemed guests, and fellow participants. My name is Khushbu Tyagi. I am PhD research scholar in the Department of Home Science at Diyalbagh Educational Institute. I am presenting my review paper that is examining the role of educational campaign in enhancing awareness regarding nutrition and adoption of sustainable nutrition practices. Sustainable nutritional practices are increasingly acknowledged as fundamentally crucial not only for the enhancement of public health outcomes but also for the promotion of environmental sustainability in a world of facing significant ecological challenges. In this context, educational campaigns assume a pivotal role in not only raising awareness among the populace but also in shaping societal attitudes and ultimately encouraging the widespread adoption of these essential sustainable practices. This review meticulously explores the effectiveness of a diverse range of educational interventions, conducting a thorough analysis of their impact on the dissemination of knowledge as well as their capacity to engender meaningful behavior change among individuals and communities. Introduction. Educational campaigns function as a crucial instrument for promoting sustainable nutrition practices. These initiatives are designed to enhance public understanding and knowledge, thus affecting behaviors and encouraging the adoption of healthier, more sustainable dietary choices.
Please be focused on your objectives and your results and conclusion of your work because time is going very.
Objective of my study are to assess the effectiveness of educational campaign in enhancing public knowledge of sustainable nutrition practices. Next objective to evaluate the impact of these campaign on the adoption of sustainable dietary habits. Third objective is to identify the key challenges and barriers to the success of educational campaign in promoting sustainable nutrition. And the last objective is to highlight successful strategies used in educational campaign and suggest areas for future research. Concept of sustainable development. Sustainable nutrition encompasses a comprehensive framework for dietary practices that not only enhance individual health outcomes but also promote environmental sustainability whilst simultaneously fostering social equity among diverse populations. Role of educational campaign in promoting sustainable nutrition. Enhancing knowledge. Educational initiatives serve an essential function in fostering awareness regarding the tenets of sustainable nutrition by circulating information through diverse mediums such as social media platforms, community outreach programs, and educational programs. Within school, these initiatives seek to elevate public comprehension of the ways in which dietary selections influence both individual health and ecological sustainability. Influencing behavioral change. In addition to the transmission of knowledge, a paramount objective.
Time is over. Conclude within 2 minutes.
Challenges in implementing educational campaign. Although educational initiatives possess considerable potential, they frequently encounter substantial obstacles that may hinder their efficacy. strategies for successful educational campaign, tailoring message and audience engagement, multi-channel communication. Future direction and research gaps. While considerable progress has been made, there remain several areas where further research is needed. These include the long-term effectiveness of educational campaign in sustaining behavior change, the role of policy support in campaign success, and the impact of emerging digital tools and platform on public engagement. And the conclusion is educational campaigns are a vital tool in promoting sustainable nutrition practices by enhancing public knowledge and influencing behavior. By employing tailored messaging, multi-channel communication, and interactive engagement strategies, these campaigns can effectively contribute to the global shift towards more sustainable food systems. Continued research and innovation in this area are essential to maximize the impact of educational efforts and support the transition to sustainable nutrition. Thank you, ma'am.
Thank you, Beta. So the next speaker is Mr. Santosh. Santosh.
Yes, madam, present, madam.
Okay. So I'm announcing few names. If these speakers are available or participants are available, then please mention your name in chat box till the Santosh is starting his presentation. So Ms. Shikha Singh, Mr. Ram Gopal, Dr. Alka, Ms. Sujeta Yadav, Dr. Sumay Roy. If you are available, so please mention your name in chat box if you are available for presentation. Santosh, please start your presentation.
Of Food Science, PhD scholar, University of Horticultural Sciences, Raichur. Technological advancement. It is a review paper. The topic was technological advancement in precision nutrient or food production. Why precision nutrient means the application of a nutrient in overall in field it is very drastically it is increasing, so that the outcome is also nowadays it is affecting the human health also, so that the precision application of nutrient in the overall field it is very crucial, and it also save the soil health.
Santosh, your slide is not moving.
Yeah, madam. Agriculture. Yes, ma'am. Agriculture is the backbone of Indian economy and water is the heart of the agriculture. Introduction to the micro irrigation. Micro irrigation is the application through the water through the drip and somewhat sprinkler and through at the root zone of the plant. Irrigation giving the irrigation to the plant at the proper time when the plant requires the water. Necessity of irrigation, and the irrigation is going to the plant when the water is not available to the plant and the moisture plant show the stress, it improves the fruit quality and plant health. It is very important for the successful production of the fruit crops. Types of irrigation. These are the irrigation types: surface and subsurface, sprinkler, drip irrigation. But majorly I am discussing drip irrigation system because of the from this drip irrigation we are applying the fertigation nutrient to the generally very precisely in nutrient application. These all are the surface irrigation. Subsurface irrigation, it is the surface irrigation. Applying water to soil directly under the surface moisture which is the plant root through. Here the water required water is large amount, so that entire field requires more water. This is sprinkler irrigation. Here the main reason this is the rotating head of the nozzle that spread the water through the.
Santosh, please conclude.
Okay, madam. Okay, madam. Drip irrigation. Drip irrigation is the major one in that we apply the fertigation through drip These are the components of the drip irrigation. When we apply the water to drip irrigation, water will apply to the root zone so that it save the water and enhance the productivity of the plant. These are the comparison irrigation methods. Advantages of the drip irrigation: saving water 50 to 60%, reduction in water loss, and also it saves the manpower, labor, over irrigation and fertilizer application. No need of drainage in case of drip irrigation. Poor quality water is also you can use that saline and alkaline water. Application of drip irrigation. These are the at farm field we are applying in our campuses. In polyhouse we are applying this one drip irrigation. Fertigation. That's earlier said that fertigation is the method in which we are applying the nutrient with the water. The main components of fertilizer irrigation is the Venturi method, sand filters, screen filter, and the pressure of the drip irrigation is 2.5 kilometer centimeter. Advantages: it inform fertilizer application, flexibility in timing of application, less fertilizer used and minimum nutrient loss, reduce cost and save labor and energy, reduce energy and demand. Disadvantage: it is not aware of any much awareness about this fertigation. Only some of the crops are following this. These all are the fertigation system. Only like grape, pomegranate, commercial crops only. Subsidy given by the RAFVAL in the government of India, 50% subsidy on Int'l irrigation. Overall, the fertigation nutrient application increases yield and net worth of land. It is helping doubling the farmer income. It increases the 20 to 30% of yield through the fertigation. Then applying the nutrient than traditional. Thank you, madam.
So this is your review work?
Yeah, madam.
Okay. So the next is Mr. Abhishek. Abhishek, please unmute yourself and start sharing of your presentation. From Himani Gautam.
Yes,
Himani.
I got a message from Abhishek that he is present and
okay,
he's joined with name
his name is
Abhishek
not
S ABISH.
ABISHAK. Okay. बढ़िया. Abhishek, now you can start sharing.
Yes, Abhishek, you are now the co-host. You can unmute and start presenting.
Okay. Mani ma'am, you have shared his message.
Good evening, ma'am. Am I audible?
Yes, you are audible. Please start your presentation.
Good evening.
Try to sum up within 5 minutes.
Abhishek, Sales Scholar, Department of Food Process Engineering from SRM Institute of Science and Technology, Chennai, Tamil Nadu. So my topic is studies on extraction and encapsulation of black cardamom oleoresin and its application. So my guide is Dr. P. Gurumurthy, Associate Professor, Department of Food Process Engineering. So introduction. So black cardamom is also known as hill cardamom. It is belong to Gingiberaceae family. So black cardamom is renowned for its own like distinct flavor and aroma. It is used as in both culinary and traditional medicine. So the black cardamom contains like essential oil and oleoresin. So the in oleoresin, there are major compounds like cineol and terphenyl acetate, which range between 50 to 60%, which is gives the aroma and flavor to the cardamom. So, but these oleoresin are unstable in environmental condition. So, I'm going with the encapsulation. So, this encapsulation of oleoresin, which is innovative process to design to enhance concentrated spice extracts. Through encapsulation, the oleoresin developed in a protective matrix, offering improved shelf life, controlled flavor release, and ease of incorporation into various applications. So, this is my work plan and methodology. So, the black cardamom is procured from an Indian Cardamom Research Center, and then it is extracted using both conventional and non-conventional method. One is oxalate and supercritical. In oxalate, the ethanol as so, and then extracted oleoresin, the encapsulation technique like with wall materials like gum arabic and maltodextrin were taken. So it is prepared using a emulsion and then using a homogenizer, and then finally the prepared emulsion is subject to spray dryer. So and then characterization studies like moisture, color, powder properties, flow properties, thermal and crystallographic, zeta and morphologies are characterized. So in my result, what I know the yield from supercritical fluid extraction, the yield of black cardamom oleoresin obtained 8.8%. So this is GCMS chromatogram of oleoresin, which contributes phenol and terphenyl acetate or major compounds, which is around 57.92% of phenol and 28.35% of terphenyl acetates. So characterization studies like stability, water activity, thermal analysis, and flow properties are characterized, and the SEM analysis of gum arabic and this is maltodextrin. So in SEM morphological, gum arabic shows like spherical and smooth surface when compared to maltodextrin. So the scope of the study is to provide a comprehensive understanding of encapsulation process and properties of encapsulated product to its potential application in various industries. So there is a rising demand in market, and there is a diverse application in food, pharma, and nutraceutical industry, and it's an innovation in growth and areas. And thank you.
Thank you, Abhishek. So, any question from audience? From co-chair or rapporteur? So the next presentation is of Dr. Shashi. Please start your presentation. Dr. Shashi, please unmute yourself and start your presentation.
Dr. Shashi, you have to unmute yourself. Yes. Doctor, you're not audible.
I think she is not audible. He or she is not audible.
She's not audible.
Because I can see he is already unmuted. He or she may be. So the next is
next.
Suman Roy. Is she?
We have Dr. Suman Roy, I think.
Yes, ma'am.
Okay.
Yeah, Dr. Suman Roy can present.
Dr. Suman Roy, you can present your presentation.
Is it visible, ma'am?
Yes, yes.
Yeah. Good evening. Myself, Dr. Suman Roy, working as a senior scientist at ICR NBPGR, and my presentation topic is exogenous ascorbic acid mediated drought tolerance in jute, that is Corchorus olitorius. So here three things are very important. One is the jute crop, which is not so common or known to everybody. The second one is role of ascorbic acid, which is very popularly known to everybody, and drought stress. So here you can see the jute crop. Jute has two cultivated species, Corchorus olitorius and Corchorus capsularis, and the chromosome number is 24. The left-hand side photo is the seedling stage jute, which is consumed as a leafy vegetable, and it has been assigned with the beauty of Cleopatra. She used to take this as a leafy vegetable. The middle one is the fiber crop, the standing crop, which we know that jute is used for. And the third picture, the right side one, is the fiber crop, which I'm trying to show the height. Jute can attain a height of 3.6 to 4 meters. So it is a 4-month-old crop. The crop initially faces a lot of drought stress, but in the later stress, it also faces submergence tolerance, submergence. So jute is such an important crop for some of the parts of India like West Bengal, Assam, Odisha, Bihar, and jute can be consumed, and each and every part of the crop is consumed, and it is now using, it has been used for bioethanol production, biobriquette production, definitely for bag and gunny bag preparation. It is well known. So there is very less information about the role of jute and drought stress. So what we thought, this is a basic slide where we know that drought stress can be mitigated by a plant through dehydration avoidance as well as dehydration tolerance. And among the dehydration tolerance, the circle one is the production of reactive oxygen species, the most deleterious compound which is producing in the plant is reactive oxygen species like singlet oxygen superoxides like this. So in the first slide, first experiment, we have tried to identify that whether in jutes, what are the parameters or key parameters which are responsible, key physio and morphological parameters which are responsible for drought stress tolerance. So we have measured all the parameters as well as reactive oxygen species, antioxidant enzymes, and we have seen these results. I am not going into detail of the results, but we have seen that superoxide production under drought stress is a very prominent signature for in case of jute crop. So our objective is to reduce this superoxide dismutage or ROS production through different application of different chemicals. We have used 6 chemicals like ascorbic acid, TIBA, salicylic acid, and we have seen that in the initial study, in the fourth picture, that drought along with the ascorbic acid is giving better result. So we have seen that ascorbic acid and to some extent PABA is also showing better results. And jute plants could tolerate a moisture content of as low as 6%. So it's a very important crop for drought model plants as a drought study. So here you can see after rewatering also, if you see the marked red color picture, after rewatering on the drought plus ascorbic acid, it is giving a very good phenotypic. No need to go deeper into the crop. The crop itself is showing its status. Then we have we went very deeper to the study that we have measured all the physiological parameters like membrane stability, T-bars content, H2O2 content, superoxide content, and plant height, all the parameters. So to say in a nutshell that jute crop along with, if we apply ascorbic acid exogenally, the parameters which are affected by drought can be regained through the application of ascorbic acid. So here also we know that proline mucilase is used to increase under drought stress. So we have seen, we will not go into detail of the variety and all the I have seen GRO204. These are jute varieties, OIN. Some of them are tolerant, some of them are susceptible. So from this also we have seen that mucilage, we have got on interesting in the previous slide that mucilage content is increasing in jute. So we have tried to measure mucilage and we have seen it is giving a very good result. The left side is the toluidine blue, aniline blue dye. And we have seen the mucilage content which has been very increased. So we have tried to screened several jute genotypes on the basis of mucilage content. There is a great variability among the crops. And on the basis, some of the part of this work has been, we have tried to publish in industrial crops and products, but the others, majority of the work has not been till published. Thank you, ma'am. Thank you.
Thank you so much for your work. But I want to know that what was the sample size of your research?
Ma'am, we have initially taken 200 jute genotypes for screening for drought stress tolerance.
From same species? All of same species?
All are olitorious. All are same species, not capsularis.
Okay, that's good.
Yes, ma'am. And this crop is very important in terms of, ma'am, environmental things, because if you want to replace plastics, jute is the very important crop.
Yes, I know. Yes. That's why I'm asking.
Yes, ma'am. Yes.
Thank you so much. So the next one is Visha Jain. Visha Jain. Thank you, sir. Visha Jain. Visha Jain. Visha, you are already co-host. Please unmute yourself and share your presentation. Visha. So if Visha is not available.
Yeah, Visha Jain has started sharing. Visha, you have to unmute also. Visha, you are not audible.
Visha, unmute yourself. Please mute and unmute yourself. Maybe there is some technical issue, so that will be rectified. Visha. So if Visha is not available,
I think we can move
then
forward.
next is Ravina. Ravina Yadav. Ravina, please unmute yourself. Now you are the co-host. Please unmute yourself.
Good evening, ma'am. Am I audible now?
Yeah, you are audible.
Good evening, everyone. I'm Ravina Yadav. Today I'm going to present my thesis work of masters on effect of phosphorus and zinc on growth and yield attributes of cluster bean. First of all, the cluster bean that is commonly known as guar is one of the most important self-pollinated leguminous crop which belongs to the family Fabaceae. India is considered as the center of the origin of cluster bean. It can use as a seed purpose, as a green vegetable purpose, as a fodder crop, as a green manure crop, because it can fix the nitrogen nearly 37 to 196 kg per hectare annually. Other than that, it is a cheap source of protein, moisture, energy, fat, carbohydrate, vitamin A, C, calcium, and iron. Other than that, it can also use for the reclamation of the saline and the alkaline soils. India is the world's top exporter of the guar gum, which is a major product. India has 3140 million per hectare of cluster bean area, 1.5 million ton of production, and 484 kg per hectare of the productivity. According to state-wise, Uttar Pradesh produce 14-18 ton of this crop annually over the area of 1979 hectare. Productivity in the state of Uttar Pradesh is 0.72 tons per hectare. After that, in this crop, these nutrients like phosphorus and zinc play very important role because their balance amount directly affect the growth and the yield of this crop. So, to check this, we have used these treatments in during my study. I have used these 9 treatments in which the treatment T1 shows the control and treatment T9 shows the 100% doses of both zinc and phosphorus. I have do my experiment in the field conditions in Uttar Pradesh in the RBD design, in which I have taken that 9 treatments with 3 replications. These are my treatments. After that, this table shows the results in which the effect of different levels of the phosphorus and zinc on growth attributes of the cluster bean on plant height, dry weight, number of nodules, and I have found out that treatment T9, where I have applied the 100% doses of both zinc and phosphorus, gives the highest yield of that nodules and dry weight, and even the plant height is also highest in this treatment T9, while the control treatment, where I have applied only the RDF doses, that is the recommended doses, it gives the lowest yield. The same pattern I have got for that yield attributes also. The benefit cost ratio was also highest in the treatment T9 and then the treatment T1 and other treatments. After that treatment T9, we have followed the pattern like treatment T9. After treatment T9, where I have applied 100% doses, that followed up by the treatment T8, where I have applied 50 and 100% doses. The conclusion of this my whole study is that if we didn't apply zinc and the phosphorus fertilizer in a balanced amount in our field, we are not able to get a good yield because the effect of interaction effect of this zinc and phosphorus fertilizer is antagonistic. If we apply high dose of zinc or any phosphorus or that lower dose of other one, then they will do the fixation, they will form the compound like zinc phosphide and decrease the availability of the phosphorus, which directly affect the yield of the crop. Thank you, ma'am.
Thank you so much. Any question from the audience? Chair and co-chair. So, Ravina, what is the significance of your study?
Ma'am, for this study, we can say that a farmer, if he does not apply the balanced dose of the fertilizer, he will decrease the yield of its crop by its own. These these nutrients like zinc and phosphorus, they are negatively interacted with each other. So the balanced amount of this is very important.
So what was the reason for selection of zinc and phosphorus for this study?
And because of their negative interaction effect, ma'am.
And what about other fertilizers?
We can say that the nitrogen, if we increase that nitrogen doses, phosphorus is directly increased in our crop. They are positively interacted with each other. Ma'am, zinc and phosphorus, they have that negatively interaction effect. They will affect antagonistically if we not apply them in a balanced amount.
That's so all these were the control experiment?
I have chosen that treatment T1 is the controlled one, where I have applied only the RDF doses. After that, or in all the treatments, I have applied the different doses of the zinc and phosphorus. In treatment T9, I have applied 100% dose of the zinc with 100% dose of the phosphorus.
Okay. So the next is Karuna.
Okay, thank you.
Okay. Hima Bindu. Hima Bindu is also available. Hima Bindu. Hima Bindu?
Yes, ma'am.
Yeah, it's your turn.
Yes, ma'am. Good evening, everyone. Today, my research myself is Hima Bindu. My name is Hima Bindu. I have completed my post graduation in Sri Konda Lakshmi Telangana Horticulture University in the Department of Vegetable Science, and my MSc research is on integrated nutrient management for sustainable spinach pit production. So coming to introduction, generally, as you all know that the botanical name is Vita vulgaris, variety bengalensis, belongs to the family Chenopodiaceae, and this spinach is a highly nutritious crop, and it title mine of minerals also, because it contains more nutrients and minerals. My research was done on spinach beet with using some of the treatment combinations of inorganic fertilizers such as urea, SSP, and muriate of potash, combined with the biofertilizers that are Azotobacter, phosphate solubilizing bacteria, potassium solubilizing bacteria, and Arca microbial consumption, along with the micronutrient spray, that is Arca vegetable special at 15 days interval. With this research, that results demonstrated that the 50% inorganic combined with 50% biofertilizer combination significantly improved plant growth, yield, and quality compared to other treatments, because this integrated nutrient management approach this enhanced the microbial activity.
Bindu, please move your slide and focus on your results.
Okay. So in this research, I have got the yield in the treatment 9, that is combination with 50% inorganic nutrients with the 50% biofertilizers. And also I have got the best quality yield. And coming to the effect of inorganic nutrients and biofertilizers on nutrients in soil on the spinach beet cultivation, these analyses were done after the harvest of the crop. So it increased the NPK content in the soil as well, and also it increased the micronutrients in the soil as well. So with this, I want to conclude that 50% inorganic nutrients and 50% biofertilizers gave the best result in growth, yield, and quality of spinach beet. So this integrated approach reduces on chemical fertilizers and enhances the microbial activity in the rhizosphere, improving long-term soil fertility. And also this biofertilizers promotes the based on nutrient management in spinach beet and support sustainable agriculture and eco-friendly food production. This study also aligned with the global efforts toward environmental sustainability, soil health preservation, and improved food security. Thank you.
So any question from audience? Now it's open for discussion from school chair and reporter. Himani ma'am. So what was the reason for selection of this one?
Ma'am, most of the farmers use only urea as a fertilizer, which is sometimes above recommended dose. So they are not sufficiently aware of management practices, and also use of other fertilizers and biofertilizers. So ultimately, this main causes the poor yield and soil. It deteriorates the soil health, ma'am. So keeping these facts in view, so this main motto was this integrated nutrient combination, which helps in crop production and soil fertility also.
What was your study duration?
Ma'am?
Duration of your experiments and study?
Ma'am, 6 months, ma'am.
6 months. Do you think that 6 months is enough to justify this work?
No, ma'am. For I have taken 3 cuttings, ma'am. This to this spinach bit, and I also have done some quality analysis, ma'am.
For 6 months only?
Yes, ma'am. From synopsis to ending of the thesis.
Okay. So the next one is Vishakha.
Yes, ma'am. Good evening, ma'am.
Good evening.
So, good evening to all of you. I'm Vishakha. I'm assistant professor in Galgotias University. Ma'am, may I share my screen?
Yeah, sure.
So, I hope my screen is visible to everyone. So, my topic is development and evaluation of value-added products from dragon fruit, Hyloceres undatus for enhanced nutritional benefits and waste utilization. This study was intended to increase knowledge about functional and nutritional properties of dragon fruit through value-added products. So here you can see some pictures of dragon fruit, and the product developed through dragon fruit, which I'm going to present, is was preserved product, that was jelly, and out of jelly, what was the residue? So from that residue, I developed certain byproducts, that is candy and spread. Standardization was done by 10 semi-trained panel members using 9-point hedonic rating scale, and further it was taken for shelf life study. So this is the flow chart that you can see for development of jelly. 3 treatments were done with pulp and peel ratio 1:1, 1:3, and 3:1. The main point of this study was that here no pectin or no gelatin was used for preparation of jelly. As we know, dragon fruit has low pectin content, but its peel has a very good pectin content, and pectin powder can be extracted. So in this study, peel was used in 1:1 treatment, and the first treatment one, that is T1, in which pulp and peel was 50-50 grams. That jelly had the best result, best texture, taste, as well as best shelf life. So you can see here that in treatment 2 and treatment 3 were slightly crystallized, but treatment 1 with a 1:1 ratio of peel and pulp were considered the best. Out of that residue of jelly, because we make jelly only from the juices, so whatever peel and pulp was the leftover peel and pulp of the strained jelly was used further for development of candy. So these you can see the pictures of candy, and then spread was also developed. So pulp and peel again was used for 2 treatment, 1:1, and the ratio in 3:1, in which the spread, the first treatment was considered the best, which has 1:1 ratio of pulp as well as peel. So organoleptic evaluation was done on 9-point hedonic rating scale, and nutrient estimation and cost calculation was also done. So for shelf life study, these were stored in glass jars in cool and dry place at ambient temperature for 2 months. And samples were carried out every 15 days to evaluate sensory changes using 9-point hedonic scale. So here you can see the organoleptic acceptability of jelly, where it was found non-significant at 5% level even after 2 months. Both taste, texture, flavor, appearance, color, everything was found non-significant. Then comes organoleptic acceptability of byproducts. So again, candy was found, difference between treatment and treatment 2 was found non-significant even after 2 months, as well as in case of spread, only the texture changed, otherwise all other values were found non-significant. So these are the nutrition composition of developed products. These all are very high in energy and can be given to any energy deficient age groups. Then cost analysis shows that even dragon fruit has a very high value. So the raw ingredient was high, but due to development of byproducts out of candy and spread, which is usually discarded, the cost of jelly as well as candy and spread, it gave value of commercial importance. So here is the chart for organoleptic acceptability. So all the parameters were considered non-significant at 5% level. Same happened with candy and spread. In spread, only the texture changed because of low pectin content, because all the pectin were extracted while formation of jelly. So this study shows that the best quality was found with pulp and peel ratio 1:1, and the products were highly acceptable during sensory evaluation, as well as no significant change during 2 months of storage, and it indicated good shelf life without using any preservative. Utilization of byproducts helped efficiently manage waste, resulting into increased productivity and low cost, showing high profitability for any venture. These products developed have very high acceptable sensory qualities, good shelf life, and nutritional properties, having many health benefits. Recommendation from these studies are these products can be commercialized and recommended due to high nutritional content. The jelly as well as candy and spread are suggested for children, underweight persons, and those requiring high energy. As far as future thirst is concerned, so impact of intervention of developed products can be studied for assessing their therapeutic properties. Development of various value-added products other than the present study can also be done to explore the other properties as well as other value-added products of dragon fruit. And techno-economic feasibility related to processing, production, and packaging can be explored for further commercialization of the products. So that was all about my study. Thank you very much.
So, Ms. Vishaka, do you think it's cost effective?
Yes, ma'am. It is very much cost effective because cost is comparable with the products which are commercialized in the market.
Because raw dragon fruit is comparatively expensive in comparison to other.
Yes, ma'am. But the thing is that when we use peel, then edible index of this product becomes very high. And that is why we are able to manage the cost. And again, when we utilize the byproducts that is also discarded, again, edible index becomes significantly high, which significantly enhances the productivity of all these products. And that is why the cost is efficiently managed.
Okay, so if the cost is manageable and then you can test the efficacy of that particular product, then you can also commercialize your product by
Yes,
testing.
ma'am. I'm in the process of commercializing the products.
Okay.
This is just a part of my study.
Okay. So that's good, comparatively good. If it shows some other significance, that it will be beneficial for you also.
Yes,
So
ma'am.
the next speaker is.
Thank you very much.
Okay, all the best. Rohila. Rohila, are you available? Rohila.
Yes, ma'am, I'm available.
Okay, please share your presentation. Till then, I'm announcing the sequence of presentation. Next will be Nishtha, Akanksha, G. Vinay.
Akanksha has already presented.
Akanksha has already presented. Okay, no issue. Then next will be Nishtha. If anyone is willing to present their work, so please raise your hand so that I can make you co-host.
Recording in progress.
So if she faced some technical difficulty, then Nishtha, you can start your presentation.
Yes, ma'am. Am I audible?
Yeah, you are audible.
Okay. Good evening, all. I am Nishtha Shikarai, and I'm going to present my topic, short-term forecasting of ginger price in Kalimpong district of West Bengal. So this is my abstract. When we talk about ginger price forecasting, we are talking about a specific spice crop, and this is a very important crop in district of Kalimpong, where we get a high quality of ginger, and it is also being exported to other states, where 60% of it is being supplied to other states. And along with Kalimpong, together with Sikkim, we contribute about 15% of the total ginger production in India. So why we are doing this price forecasting is so that we can know what we can do in the times where the ginger is not produced. So that is usually during April to June. So my study area, I have taken time, the data that I have taken is from August 2023 to March 31st. So for after 31st March, I will be forecasting. This study is done by dividing the set into two sets, that is training and testing, 80 and 20%. And then we will I have used some techniques to develop the forecast. So I have used these three techniques. These are the review of literature. One is the ARIMA model, the other one is the time delay neural network model, and the last one is the hybrid, which is a combination of both the models. Univariate Box-Jenkins ARIMA model, it is the primary form of time series model, and it is the very basic statistical time series model. This is the first one that I've used. Secondly, which it comprises of 3 steps: identification, estimation, and diagnostic checking. We identify the parameters, then we estimate the parameters by checking which one is the better of all the models, and then we go on for a diagnostic checking of all these. Then the second model is a time delay neural network, which is a machine learning model, and this is very famous for its use in systematic sequential data. The third is the hybrid model, which is a combination of both, where the residues of the ARIMA model is used to and combined with the time delay neural network. Okay, so the first thing that we saw is that from the ACF plot, we saw that the series is non-stationary, and also from the ADS test, we found that it is non-stationary. So we differenced it, and then we found it as it is stationary. From the p-value, we can see that it is significant. So at d is equals to 1, it is significant. So it means that at differencing one order, it is stationary. Then we identify the ACF and PSF plot. From there, we identify the orders. We see at lag 1 and 1, ACF and PSF, they are both significant. So based on that, we will we make models, and from there we identify which model is better. So 0, 1, 1 without constant was better. So this is the model we have used. And these are the parameters, the MA1, the moving average model, that coefficient is 0.499. So, and we found that it is also significant. The other thing is that these residuals are also independent, because it is non-significant. The Q statistic p-value is non-significant. Then we have done to do the time delay neural network, we need to know whether the series is non-linear or not. So based on the p-values, we see that all of them are significant. So it means that it is non-linear. So we can go on for doing TDNN, as well as on the ARIMA residuals also. We found that they are non-significant. They are all significant except at epsilon 4 of m is equals to 3 dimension, but majority of them are significant. So we significant. So we conclude that it is suitable for hybrid modeling. In case of both the models, hybrid as well as time delay neural network, we have taken 8 input nodes and 6 hidden nodes.
Esther, please conclude. Please conclude.
Yes, ma'am. Yes, ma'am. Okay, time delay neural network and hybrid model. Then after evaluating all of these models, we found that the hybrid model, which is a combination of both, was the better one. So by looking at the residual plots also, we can see that they are all, they do not have significant lags, and which means that all of these models are good. Then on the basis of the last models, we see that they are having, they do not change so much, like only in the first and second week of April and third week of April, there is a slight change, but then onwards, there is a constant. So what we can conclude of this thing is that we have found some models and then we have forecasted them, and the price forecasting was done for the time period of 5 weeks, that is from April 1st week to May 1st week. So during this period, we saw that there is not much change, and this is a very good result, so that the farmers they do not have to be so anxious about this period of time. Maybe after this, there might be some changes, and during that time, they will have to look for other options. Thank you.
So this is your review work or original work?
Original work, ma'am.
Or survey type of work?
Yes, ma'am.
You have selected a few sampling sites or collected the data from several other sources?
Ma'am, collected data, it is collected data from agmarknet.
Okay.
Secondary
Thank you
data.
so much.
Thank you.
So, Raichur, please stop presenting your presentation. Ruhila has already started. So the next will be Sadhna after Ruhila. Sadhna, be ready. And Visha Jain, next is yours. Visha is ready for her presentation. So first is Ruhila, next will be Sadhna, and next after Sadhna, Visha Jain. Ruhila, please start. Please unmute yourself and start your presentation. Ruhila, am I audible to you? Okay, I'll try to unmute yourself. You can try to unmute yourself.
Ma'am, Ruhila is not co-host.
I think I have removed her. I'll check. Yes, now you can unmute.
Hello, ma'am. Am I audible?
Yes.
Good evening, participants and scientists. I'm Dr. Rohila. I'm from Kashmir. I have completed my doctorate in soil science and agricultural chemistry. I am here to present a poster on the title Evaluation of Suitable Extractant and Determination of Critical Limit of Nitrogen in Onion under Temperate Conditions of Kashmir Valley. So in this this was basically my study in PhD degree program, some part of it. So in this I have selected 4 extractants, namely alkaline permanganate, potassium chloride, hydrogen peroxide, and potassium dichromate. I was of the opinion to see which extractant was suitable to extract nitrogen from onion growing soils. And for this study, I correlated this various extractants with braze percent yield and an uptake by onion. And after that, alkaline permanganate was found to be a suitable extractant. And for the critical limits of nitrogen in onion growing soils and plant, it was found to be 243 ppm for soil and 0.2% in 25% in case of this onion bulbs. Coming to materials and methods for this study, I carried out pot culture experiment. For that, I selected 15 locations of onion growing soils where I collected samples. After processing of the samples, I put this processed soil, 4 kg soil was put in the pots, and 2 onion seedlings was grown in each pot. And different after this harvest of the crop in the month of July, different chemical analysis like chemical extractants, which I already said, were used for the extraction of nitrogen. The experimental design, it was a completely randomized design. Locations already discussed were 15, and different levels of nitrogen like control 50, 100 and 120 kg per hectare were used. Number of replications were 3, and the total number of pots used were 180. Result portion, after the harvest of the crop, different like extractants that were used were correlated with braze percent yield at 1% level of significance and an uptake. And it was found that 30% hydrogen peroxide and alkaline permanganate showed a highly significant correlation with braze percent yield and this an uptake. Compared these two, hydrogen peroxide and alkaline permanganate, it was alkaline permanganate that showed highly significant correlation. And the order of suitability of the 4 extractants was alkaline permanganate, hydrogen peroxide, and potassium dichromate, and 2 normal KCl was least this suitable extractant in case of this soils in Kashmir. Now coming to critical limits of available nitrogen, that were calculated using scatter diagram given by Cat and Nelson. So critical limits of nitrogen in soil found out to be 243 ppm and 0.2% in case of onion bulbs. Conclusion: the correlation between nitrogen extracted by different methods and braze percent yield and nitrogen uptake revealed the best method of assessing available nitrogen is alkaline permanganate, and 243 ppm nitrogen by alkaline permanganate was established by Cat and Nelson technique. Below this, by this critical limit, we mean that below this critical limit, the plant will respond accurately to the fertilizer applications. Thank you.
Thank you, Rohila.
Thank you, ma'am.
Any question? Rohila, what was your sample size?
Ma'am, it was 20. I selected 15 locations, onion growing soils.
15 or 50?
15, 15, ma'am.
15. And
Yes,
how many samples you have collected?
Ma'am, I collected 15 samples only. I collected more than 15 samples, and then finally I selected 15 samples for the spot study.
But I think it's a small sample size for this type of study. You have to do widely. The work is good, but I think sample size is small. But
Ma'am,
you
actually,
stop
we
adding
have to select only, ma'am, actually, we have to select only commercialized areas where this onion was growing commercially. That is why it was only 50 locations where we found that onion was commercialized. That is why.
Okay, there are only 15 locations, those where the onion has been.
Yes, ma'am. Yes, ma'am.
Okay, that's the reason. But you can increase your sample size You can select the locality. Say locality will be fixed as per your choice if you are planning for any research. But
Yes,
you can
ma'am.
increase the particular type of samples.
Yes,
So it
ma'am.
will be comparatively better for our
Yes, ma'am.
researchers.
I'm thinking about to write a project where I will improve the sample size.
Okay, okay. So the next is
Thank you.
all the best. Sadhana.
Ma'am, I'm audible?
Yes, you are audible.
Ma'am, my screen is can you see my
Your
screen?
screen is not visible. Your name is visible on the screen. No. Now it's visible.
Is it visible, ma'am?
Yeah, yeah. Now you can start your presentation.
Okay, ma'am. Good morning, good evening, ma'am. Good evening, all of you. This is my research topic, mapping the green invaders, diversity and dominance of weed flora in okra ecosystem. Actually, my work was to find out different weed flora and to find out different uh, weed diversity, weed control efficiency by different weed management practices. This A comprehensive study was conducted on weed flora in my experimental field to unveil diverse and dynamic weed spectrum comprising all grasses, sedges, and broadleaf weeds. The most commonly found sedge was Cypress rotundus. It was more dominant, and grass was Cynodon dactylon, and many broadleaf weeds were found. And we can go with conventional method and also the innovative methods. The conventional method I used was different pre and post emergent herbicides and different plastic mulches and also straw mulch and different next like intercropping with palak and stale seed bed, going with hand and mechanical weeding. And my experiment was conducted in Arka Anamika variety of okra in College of Horticulture, Rajendranagar, SKL TGHU. There were many weed flora found in my field. They were enlisted category wise, and at 15, 30, 60, and 90 days, they were in different, in each plot, 5 different places I have collected and then dried them, and I have took the weed density, weed control efficiency, and weed index was calculated. Out of this, the best result I have found that black polythene sheet mulch gave the best results. Like maximum, minimum weed flora was there. Almost it is only sedge was found. No other broadleaf weeds and any grasses were not found. Those sedges was also due to they sedges have the characteristic features like they have sharp shoot tips, and rhizome growth was very faster, and they can germinate even under darkness. Mainly in okra, these black polythene sheets are very much preferred because basically okra is tropical and subtropical, and it requires warm climatic conditions. So black polythene sheet mulch just absorbs sunlight and increases 1 or 2 degrees soil temperature when compared to outside temperature. So maximum weed control can be seen with black polythene sheet mulch, and it is on par with simultaneous hand and mechanical weeding. But when it comes to economics, I prefer only black polythene sheet mulch. So different herbicides are also used, but these herbicides have a residual effect, so I don't prefer herbicides also. So I conclude by saying that farmers must use black polythene sheet to control maximum weeds in okra fields. Thank you, ma'am.
Thank you. Then, what is the reason for selection of black poly?
That's what, ma'am. Okra generally
in okra, maximum yield losses will be there, ma'am. Up to 80 to 90% yield losses will be there. So we need to any number of times, if we are going for hand weeding or mechanical weeding, it is very costly. So it is better to go for black polythene sheet mud. It is economical and as well as proper with almost 99% weed will not be there with black polythene sheet.
Okay. So the next is Visha Jain. After Visha, Vinay, and after Vinay, if anyone is willing to present oral presentation, please mention your name in chat box. Visha, start your presentation. So, Manas Ranjan. Manas Ranjan, your presentation. No, I think you will be presenting poster. So, next is Vinay. Vinay, please present your work. Participant, please wait for oral presentation, and for oral presentation, then we will start the posters again because these are already in queue.
Good evening, ma'am. Is I audible, ma'am?
Yeah.
Good evening to everyone. Myself, Gaddam Vinay, research scholar from Department of Forestry, Wildlife and Environmental Science, Guru Ghasidas Central University. And my title presentation is Morphological Characterization of Dalbergia Siso in Chakarbata, Bilaspur and Chhattisgarh. Introduction of my topic is Dalbergia Siso, also known as Indian rosewood or sesame, offer various benefits including medicinal properties and timber use. The medicinal property main used from the tree bark, leaf, and hardwood are used to treat conditions like fever, skin disease, piles, and ulcers. It also used for its anti-inflammatory blood purifier. Its leaves can be used as fodder, and its wood is valued for furniture and construction. The tree also play a role in traditional medicine with various parts. The study area location is Chakarbata, Bilaspur district, Chhattisgarh. Climate tropical, annual rainfall 1100 millimeters, soil types sandy loamy and clay loamy. And methodology of this topic is field survey is conducted during the growing the season in 2024. Sample size 30 matured and healthy tree randomly we selected, and trace we measured the tree height, DBH, crown spread, leaf morphological, and data collection tools will be measuring tape, calipers, field data sheets, GPS. And morphological traits that we observed, tree height will be 10 to 22 mean, and GBH range will be 40 to 90 centimeter, and crown spread will be a range 4 to 8 millimeter. And leaf character, leaf length is the 18 to 14 centimeter of different trees, and leaflets per leaf is typically 3. Shape is ovate to broadly ovate. The result that we observed the variability, high phenotypic variability among individual trees, and significant difference in tree height and crown architecture. Variation in leaflet shape and size indicating adaptively to micro environmental. Thank you.
Any questions, query? Any questions from audience or co-chair? Any question from co-chair? Okay, thank you so much, Vinay. So next
Thank
is
you.
Rajshri. Bhagwat Kumar. Bhagwat Kumar for oral presentation. Please show your identity again so that I can unmute you. So Rajshri, please start presentation. Redmi, please change your name so that I can identify. Bhagwant Kumar, please change your name so that I can identify you.
Ma'am, please make Rajashri co-host.
Ma'am, please repeat.
Yeah, she has raised her hand.
Yes.
Dear participants, if you are unable to change your name, then you can also raise your hand.
Yeah, so that we can add you as a co-host. Okay, now I can identify. Rajshri, please start your presentation.
Good evening, ma'am. Can you see my presentation?
Yeah.
Okay. My name is Rajshri Krishna Bhargava. I am an MSc scholar, Department of Agronomy, Banaskantha University. And this is my MSc research topic where I'm doing the evaluation of high value and climate smart crops under custodial-based agri-horticulture system in the Vindhyan region. And the research question that I'm addressing is how to deal with climate change and food security challenges in present century and the probable solutions of crop diversification, cropping sequence, agroforestry, and climate resilient agriculture, along with dryland agriculture and agroforestry. So these are my contents: introduction, methodology, results, discussion, conclusion, and way forward. So, in introduction, Dr. P.K. Nair has defined agroforestry as a land use system that integrates trees, crops, and livestock in a manner that is scientifically sound, economically beneficial, and practically feasible. According to a research of Ramamurthy speaks of bilateral cooperation. So, based on that, there are two objectives in my research: to assess the productivity, profitability, and resources efficiency of different agri-horticulture systems, and to assess the economics of the treatment. These are the photographs of the various treatments. The methodology was that the treatment was made in RBD design in sandy clay loam soil of Rajiv Gandhi South Campus, Banaskantha University. This is the details of the treatment. There were 5 treatments of okra, cowpea, cluster bean, fingermillet, and ashwagandha. The varieties were okra VNR 999, Kashi Kanchan, RGC 936, VL 352, and CIM Push for ashwagandha. So these are the various components of the experiment: agroforestry, crop diversification, dryland agriculture, sustainable agriculture, agri-horticulture system, climate smart agriculture. The results showed that the mean crop equivalent yield was highest for okra followed by cluster bean, and it was lowest for finger millet, and the BC ratio was recorded highest for finger millet and the lowest for cowpea under custard apple. In conclusion, I would say that such type of research can help in addressing the sustainable development goal, like sustainable development goal number 15, life on land, SDG 2, zero hunger, SDG 13, climate action, SDG 3, good health and well-being, SDG 11, sustainable cities and communities. And these can also address the RED plus activities like carbon sequestration and conserving biodiversity. So the way forward is that we are trying to address the trilateral cooperation where US identifies that India has great scope in trilateral cooperation, especially in climate smart agriculture and renewable energy, SDG, and meeting food security demand by 2050. It refers to a trustworthy solution. And this is the flowchart for trilateral cooperation. These are the references I use. Thank you.
Thank you. Rajshri, what do you mean by RED activity?
Yes, ma'am. REDD plus means reduce emission for deforestation and forest degradation. It was actually introduced in India in 2014, but after that it changed to RD. First it was RED, then it became REDD, then it became REDD plus. Now it is a triple win concept. We are addressing the biodiversity loss. We are addressing the upliftment of the rural communities. At the same time, we are addressing the food security. And such experiment, I think, can address this RED plus in future.
Okay. So next is I think there are few presentations. Bhagwat Kumar. Bhagwat Kumar. Bhagwat Kumar. I have already added you.
Yeah, he's co-host.
Yes, ma'am, I have already added. Yeah, joined with his
Participants, without wasting much time, you can just start presenting.
So there are three more presentations: Kota Karuna and Manas Ranjan and Bhagwat Kumar. If anyone is willing to present, please raise your hand or mention your name in chat box. Bhagwant Kumar. So next is Manas Ranjan. Manas Ranjan. Mehraj Sophie. Okay. Manas Ranjan, please present your work.
Yes, ma'am. Ma'am, is it audible?
Yeah, or visible.
Yes. Good evening, everyone. I am Manas Ranjan Pereda, PhD scholar of Department of Horticulture Science from Dr. Rajendra Prasad Central Agriculture University, Pusa, Bihar. I am over here. I would like to present here on the topic unlocking the nutritional potential of onion leaves, impact of sowing dates on bioactive compounds. Nowadays, we are more reliable the sustainable agriculture for the food production for food security. But now my crop was onion, and mostly onion is mostly grown for the bulk production, but in spite of unknowingly leaf are destroying. But now the people are more potential adapting for the onion leaf consumption. So due to their high nutritional value. For this, my experiment was done at a regional research center and technology transfer station, Bhawanipatna, Kolhandi, Odisha. And this is my MSc thesis research work, and my design was the randomized block design, and treatment was 5 and the replication is 8. 4. And this is my the treatment details. D1, which denoted as a 15th June swan crop, which was transplanted 60 days after the sowing, that is 15th August, and lichen. And
Please
this
move
is
your
the my.
screen. I think it's a
Pardon?
part of your abstract. Your part of your abstract is visible on screen, but not your poster.
So,
A
ma'am,
part of your poster is visible.
Kindly zoom in. You have zoomed it out so much that we are not able to see it properly. Or you can put it on full screen mode. The fourth option, which is slideshow option, you can go for that. Yes.
Yes.
This is the slide mode option.
So please continue.
Yes, this is my the experimental design, and here I found that the early swan crop, which was transplanted during the 15th August, which was a high mortality rate, which is less survival rate. It is due to the high rainfall during the monsoon season, and according to plant height, number of leaf per plant, and leaf length diameter also is high during the early season late sown crop as compared to the early sown crop. In spite of that, the early sown crop has a better nutritional quality of a point of view, that is vitamin C, ascorbic acid, protein, and other nutritional point of view was found in the better in early sown crop. And in coming to the yield point of view, marketable bold yield was found in the late sown crop, which was better in as compared to the early sown crop. And here we can conclude that if we will go for the throughout the year planting, then late sown crop provide the better yield as compared to the early sown crop. But the early sown crop is really high nutritional value as compared to the late sown crop. Thank you, ma'am.
So the next is Kota Karunashri. Kota Karunashri. Kota Karunashri. Okay, I'm meeting host. 1 minute. Okay, now you can unmute yourself. Kota, you are now co-host. Now you can unmute yourself.
Good evening, ma'am. Is it appearing, ma'am, my screen?
Yeah. Next is Mehraj Soni. Mehraj Sophie. Okay, please conclude within 5 minutes.
In the world where climate shifts are no longer predictions but daily realities, food on every plate is now a question of survival, especially in South Asia. Good evening. I'm Karuna from UAS, Bengaluru. I'm here to present my poster title named Climate Change and Food Security, a Vulnerability Assessment for South Asian Countries. We all know food security is a persistent issue globally. And in South Asia, it is particular. It is vulnerable, with more than 800 million population are undernourished. Climate change combined with the population growth and water stress threatens agricultural productivity, food availability, especially in 25 dependent economies. Most of the studies address either climate change or food security. Now here, I am here to combine climate change and food security and how it affects the food security regarding climate change. Regarding my significance of study, it focuses on regional and says about the global impact. It explains direct link between climate change and food security and country-wise assessment of the climate change impact on food security. So here is my objective: to assess the vulnerability of South Asian countries to climate change in relation to food security through composite vulnerability index. So this is my study area. Around 8 countries I have selected. Why I have selected South Asia is 60% of people are involved in agriculture. 60% of the people involved in agricultural labor and 20% of GDP contributed around the economy in the South Asian countries. So my methodological process, I have collected the data from World Bank. So I have collected climate indicators. I have collected world development indicators. So coming to that methodological process, I have run principal component analysis. And I have constructed an index which is called composite vulnerability index. So coming to the results and discussion, let me say what are all the variables I have selected. Exposure. Exposure means exposure is the nature and degree to which system is exposed to significant climate change. Here in the context, how food security is impacting or it is exposed to the climate change, how it is related. So for that, I have taken maximum, minimum temperatures, precipitation, rainfall. If we can see here, Afghanistan is more moderately, it is exposed and indicating susceptibility to temperature fluctuations. Coming to Nepal and Bhutan with a low exposure and relatively less temperature fluctuation extremes. So coming to the sensitivity we know how system is exposed or responds to the changes in the climate. So Bangladesh and Bhutan, Bangladesh has moderate and it's have highs in index number, and Bhutan has very low. So here, Pakistan, Bangladesh are moderately vulnerable to the climate change. So they depend on agriculture potential vulnerability and food production disruptions. So here coming to adaptive capacity, how the system is cope up with the climate change and how it can overcome the vulnerabilities. So here in the adaptive capacity measures, I have taken GDP per capita, road density, rural population, unemployment, poverty, headcount ratio, etc. So the adaptive capacity is higher for Maldives, suggesting good infrastructure and socio-economic conditions. And if we can see, Afghanistan and Nepal has low adaptive capacity. So coming to the overall vulnerability index for the South Asian countries, Afghanistan is highly vulnerable.
Karuna, please.
Yes, ma'am. Vulnerable, followed by Bangladesh. So if we can see here, this is called spider map. So it is 0 to 1. Here, if it is coming out to the map, they are very vulnerable. So adaptive capacity is inverse. Maldives has highest, so it is showing likewise. So coming to the policy making, we have to invest climate resilient agriculture, education, and infrastructure, especially in Afghanistan, Pakistan, and Nepal. And also the drought countries, we should introduce efficient irrigation and rainwater harvesting systems to reduce the sensitivity in drought-prone areas. Here is my reference. Thank you.
Thank you, Karuna. You have collected data from third-party sources.
The data is completely secondary, ma'am. From World Bank, ma'am.
From World Bank you have collected, and what type of analysis you have studied?
Yes, ma'am. First, I have to conduct correlation and which variables are significant. Then I have to run principal component analysis, ma'am. And then I have to take that weightage through eigenvalues. We have to take weightage, ma'am. And then from that we have to construct index, ma'am. It is from 0 to 1. If it is near to 0, the values which are near to 0, so the countries are very less vulnerable. If it is near to 1, the countries are more vulnerable to the climate change and food security.
Okay. So you have collected the data from their website or some other resources?
Yes, ma'am. I have collected data from World Bank, ma'am, from 1950 to 2023, ma'am.
23.
Yes,
For
ma'am.
24 and 25?
No,
For
ma'am, it is not yet
30 and
updated, ma'am.
for 24? The data is available or not?
No, ma'am, it is not
Okay,
updated.
it was the last updation.
Yes, ma'am, yes, ma'am.
Okay. So the next is Pramod Kumar Mishra. Pramod Kumar Mishra.
जी मैम.
This will be Mehraj Sophie.
Hello. Ma'am, I am audible.
Yes, you are audible and your screen is also visible.
Good evening, ma'am. I am Pramod Kumar Mishra from the Department of Entomology, CSUT, Kanpur. My research focus on screening of chickpeas, some promising genotype of Bengal gram against Bilaspur.
But your screen is not clearly visible. I think when you are preparing your poster, it should be visible. Because you have already put it so many content in your slide. You should capture only few and it should be point to point.
Okay.
Okay, no issue. Please present your work.
Okay, thank you, ma'am. Chickpea is considered as king of pulses and also known as Bengal gram, CC or garbanzo beans. Pulse beetle is a key pest of storage since chickpea is a major protein source. Protecting it during storage is a crucial to reducing post-harvest losses. The objective was to evaluate the susceptibility of 22 chickpea genotypes to Plasmodium and identify resistant lines for future breeding. We observed egg laying, number of adult emergence, mean development period, growth index, and weight loss over 30 and 50 days after insect release with the following formulas like adult emergence is equal to number of adult by total number of egg laid into 100, mean development in number of adult emerge on a day into number of days taken for adult emergence divided by total number of adult emerge, adult mortality is equal to number of dead adults by total number of adult released into 100. Growth index percent adult emergence by mean development period into days. Percent weight loss, initial weight of grains minus final weight of grains divided by initial weight of grains into 100. So we have found maximum number of egg laid in CL7 genotype CL7 and minimum number of egg laid in CL21. And the adult emergence is maximum seen in CL7 and minimum in CL13. And the adult emergence percent are maximum in CL16 and minimum CL8. These are the genotypes CL series CL1 to CL22. And the maximum mean development period are seen in CL8 about 29 to 35 days and minimum in CL9 about 21 days. And the growth index is maximum in CL4 and minimum in CL18. And weight loss are maximum seen in CL4 and minimum in CL18 genotype. So, genotype so conclusion several genotypes like CL8, CL13, and CL18 are so promising resistance against Sclerosobrucus sinensis and highly susceptible varieties are CL18 and 15 and 18. Thank you, ma'am.
Thank you, Pramod. So the next one is Mehraj Sophie. After Mehraj Sophie, next presentation will be of Bhagwant Kumar as he is interested to present his work. So if anyone is willing to present his or her work, please mention your name in chat box or raise your hand so that we can make you co-host and you can start your presentation. Bhagwant Kumar, you are already added as a co-host. Mehraj Sophie, unmute yourself and start your presentation. Mehraj Sophie.
Yes, ma'am. Am I audible?
Yes, you are audible.
Ma'am, I'm going to share my screen, but I think a little bit technical
No,
issue.
if you are finding some technical difficulties, then you can share later so that Bhagwant Kumar can present. Okay?
Yes, ma'am.
Okay, Bhagwant, please share your presentation. Please rectify your technical issues so that your presentation will be last one. Bhagwant Kumar. Bhagwant Kumar, please unmute yourself and present your presentation. Okay, we can wait for 2 to 4 minutes so that all the technical issues can be rectified. So if you are unable to rectify, then you can present tomorrow. Okay.
Bhagat Kumar, kindly unmute and start presenting. Otherwise, you will lose the chance to present as we are going to conclude the session.
Good evening, ma'am. Good evening to all of you. Can I audible?
You're audible and you are required to share the screen. Yes.
Maharaj Sophie, stop sharing so that Bhagwant can share his presentation.
Maharaj Sophie, you can just do the required settings. Till then, we are going to have the presentation of Bhagat Kumar. And up to then, you can just make all the required preparations. You can just start your PPT and then you have to present.
Bhagwant Kumar, please unmute yourself and start your presentation. If you are not presenting your work, then you can present tomorrow if they allow you, because as per the schedule, your
My
presentation
screen is
schedule
visible, ma'am.
no, your screen is not visible. Your voice is audible.
I think you have joined from your mobile phone. If you are unable to present using your mobile phone, you can join in the tomorrow's technical session with
Okay, thank you.
with the laptop so that you can present.
Okay, thank you, ma'am.
Yeah, sometimes there is a problem with mobile phone.
Okay, thank you.
So next one is Mehraz Sophie.
Mehraz Sophie, you can try with your presentation. If you are able to present, then you can present.
Yes, ma'am. I am trying a little bit.
Yeah, if there is any issue, you can present tomorrow also. Yeah, Bhagwant Kumar has started presenting.
Hello.
Hello, yes, you are audible and your screen is also visible.
Okay. Good evening, ma'am. Myself, Bhagwat Kumar, Assistant Professor Scientist from Post Harvest Technology, Horticulture and Research Station, Jagdalpur, Bastar. Here I am presenting my project work. The title is Bastar Tribe Women Empowerment and Sustainable Livelihood through Secondary Horticulture. Secondary horticulture is a conversion of horticulture produce in various produced and served to the consumer. Here my abstract is we are develop model for Western tribal women for livelihood, sustainable livelihood. Here we train for the processing of turmeric, chili, coriander, and pickles. Here we are develop various kinds of processed product like turmeric powder, chili powder, jackfruit pickles, jimmican pickles, keukan pickles, and these produce are sold in various tribal ashrams,
Madam, I think he's facing some technical issue because he has left from the meeting. So I think Mehraj Sophie can try to present if he is able to present.
Ma'am, he has sent his message that he is not able to share his screen.
Okay,
So
okay,
he will be presenting tomorrow.
Okay, then I think we can keep his session on tomorrow and also for Bhagwat Kumar because I think there is some technical issue with him.
Yes. So we can conclude the session.
Yes, we can conclude the session and these two participants we can have their presentation tomorrow.
Yes.
So
So the last of the session, I would like to thanks to all participants, my co-chair, Dr. Asha, and the reporter, and Himani ma'am also for her patience and Jaya ma'am, she was also available during the event, and also to the Jiten sir, he was always available in all the sessions. So I'm also thankful to all the participants for their patience. So out of 40 presentation, 22 has presented their work, although two were also trying to present their presentation, but due to some technical difficulties, they will be presenting tomorrow. So congratulations to all presentees and thank you so much to Jiten sir for inviting me as a chairperson of this event. Thank you, sir.
Thank you, Nisha. Nisha.
Thank you so much, sir. And also thanks to Himani ma'am, she was also available during the entire session for her patience.
Yes, yes.
Because we were performing our job, but she was being a host, she was also available.
thank you, thank you so much, and you have managed it very well. So I just want to thank all of you for joining and being a wonderful audience, and to the to Dr. Nisha ma'am, she has very patiently handled the session. Although we have lots of presentation and the participants were also facing some technical issue, but madam has managed it really well. So it's very commendable, and thanks to the participants, thanks to the organizers. And we have successfully concluded first day of this conference, and tomorrow we'll have more presentations. So till then, very good.
Okay, then good evening and
Yeah.
good night to all of you.
Yeah.
It's night time.
Yes, yes. Everybody have a great evening, and we will join tomorrow.
Okay, thank you so much.
Thank
Sir, should I make you as a co-host so that you can save the recording because I am the host of the session. I think sir has left. Sir, I'm asking, should I make you the host of this session so that you can change the settings accordingly, or should I end the session? जितेंद्र सर, मैं पूछ रही हूँ कि मैं क्या ये सेशन एंड कर दूं या फिर इसको आपको को होस्ट कर दूं जिससे कि आप इसकी टेक्निकल सेटिंग जो भी है वो कर सकें। सर मैं यही पूछ रही हूँ, मैं आपको को अच्छा 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 अच्छा। मैं आपको यही पूछ रही थी कि आपको होस्ट बना, मैं यही पूछ रही थी। बिल्कुल बिल्कुल ठीक है, मैंने कर दिया। ठीक, ओके ओके सर।